Hi, everybody. My name is Ed Hutchinson. Thanks very much for joining us today for our second online Prime webinar. And the theme today is on the similarities and distinctions between long COVID and ME/CFS. We've got 5 speakers lined up for you today, each of whom will speak for about 20 minutes, and then there'll be 10 minutes more of questions and discussion. If you do have questions or comments along the way, please do use the chat function on the Zoom platform. And we'll do our best to get through as many questions and comments as possible throughout the afternoon. I should point out a couple more things. The event is being recorded, and the recording will be made available soon, and details of which will be via the Action for ME website. And I should also point out that we'll be having a break during the program at around 3:30 after our third presentation. So the break will be about 15 minutes long. So before we hand over to Professor David Price, who'll be chairing our first session, just wanted to give you a very quick reminder of the objectives of the Prime project. Prime is a 4-year funded project by the Medical Research Council and started in October 2025. And primarily its objectives are to do with networking and facilitating collaborations. So these online webinars are very much part of that. We're trying to encourage knowledge transfer, make connections, form new research collaborations. And this work continues previous research studies, such as the Decode ME genetic study that finished last August. So principally, we're looking to form consortia and collaborations in genetics biomarkers with a view to helping the development of diagnostics and effective treatments. We've also established a Prime LinkedIn group. The link is shown at the bottom of the screen there, and we'll put this into chat later on, so it's available. And we would encourage as many of you to join that group as possible, please. Really is a forum for discussion, sharing news, developments, etc., and hopefully it'll be a really good way of disseminating information. Any questions, comments, requests for more information, please do use the email address again at the bottom of the screen there as well. And finally, our final objective is really to ensure that patient and public involvement is very much at the heart of what we do. And to give you a little bit more information on that, I'll just hand over to Sian Leary from Action for ME, who's going to take you through this activity.
Hello, everyone. I'm Sian. I work for Action for ME, and Action for ME are working in partnership with University of Edinburgh to deliver this aspect of Prime, which is the ME/CFS Research Involvement Hub. So what we're seeing across kind of medical research sector is that patient and public involvement in research, in helping to shape research, is being recognised as incredibly important and very effective at improving the research that's being undertaken. So big funders like NIHR, MRC, Wellcome Trust, Horizon Europe all require patient and public involvement for bids to be successful now for research. So we're hoping to help make that a smoother process in the ME world and support the research that's happening in the UK, at least initially, to have really strong patient and public involvement at its core. So we're at the moment piloting our processes for recruiting people. But come the 18th of May, we're really excited to say that we're going to launch recruitment more broadly to people with ME and their carers and close supporters. And we're aiming to build a hub that's got at least 100 people in it and train them to help shape research. And then, we've already got a couple of projects that we're hoping to work with, but we very much want to start promoting this research involvement hub to ME/CFS researchers more broadly. So please do get in touch with us at prime@actionforme.org.uk because we are funded for the next 4 years to try and help you deliver PPI in the best way possible for your projects. We are developing processes to help you understand how to budget for PPI, and we aim to match people with your projects who are going to be able to provide that lived experience input to research and improve things in so many ways. And I think, it's great. We've got David Price and others here who I know already use PPI in their own work, and I think they see the benefits of it. So we are really excited to help make this the norm for research across the ME/CFS sector. Where if you're a person with ME or with a close one with ME, then please look out for that launch on May the 18th. It'll come through Action for ME as we're coordinating it, and we look forward to getting you involved. Thanks very much, and I'll pass over to David Price now, who's going to host these next talks.
Thanks very much, Ed, and thanks very much, Sean. Just to say from our perspective, PPI is critically important and has been hugely enabling for our work. So it's great to see that it's at the heart of Prime. So in the first session, we have three outstanding speakers. First up is Danny Altman. Danny is one of the godfathers of immunology in the UK. And he's also been a leading scientific light for all of us throughout the pandemic and in the post-pandemic era. He's currently a professor of immunology at Imperial College London, and he's going to tell us about his new program on post-infectious syndromes, particularly long COVID and ME/CFS, which is entitled Rosetta Stone. Danny.
Thank you. Let's share. So I'm hoping that's okay. So this is called Immunological Mechanism in Long COVID and ME/CFS, a Rosetta Stone to decode post-viral sequelae. And I come from Imperial College, and you can find me on Blue Sky, as shown there, no longer on Twitter or X or whatever it's called now. And those are my disclosures. So forgive me for a moment, I really kind of labor the metaphor of how this became Rosetta Stone. Just to say that, you all know what the Rosetta Stone is, but the whole point is, it seemed apposite because here you had people who simply had a language that couldn't be decoded, but through being able to compare different scripts, hieroglyphic, Demotic, and Greek, in 1822, it got decoded, and this was an incredible breakthrough. And so we started to refer to the Rosetta Stone here as the way that our three parts to the tablet would be ME and long COVID and healthy controls who make a complete recovery from their viral infections. And for reasons that I promise you will become much clearer as we get through the 20 minutes, it kind of makes sense, and I think hopefully will pay off. So like David very kindly said, we were one of the labs among many others that were working hard during the pandemic when the heat was really on, wasn't it, to work out what on earth was going on with this new virus. And we, without getting into it here, we played some part in contributing to a lot of that story about effective immunity to SARS-CoV-2, and published a load of stuff in places like The Lancet and Science and Nature on T-cell and B-cell immunity and kind of the rules of engagement in the emerging response to that virus. And somewhere, again, as many people here know, at least as well as I do, if not better, somewhere around that period, about the summer of 2020, we were all beginning to have meetings with people and have friends, colleagues, whatever, who'd had their SARS-CoV-2 infection and simply hadn't returned to baseline health and were variably unwell, some of them incredibly unwell, and some of them unable to come back to work. This is a graphic from an artist called Monique Jackson, who kept a kind of online diary of this on Instagram and supplied a lot of the artwork for long COVID support. So we in the lab were starting to get drawn in, not just to the world of acute COVID infection, but long COVID. And a sort of, a confession, admission, whatever, if you'd asked me at this stage in 2020, 2021, my understanding as a researcher of ME was practically zero. And I had done no research on ME. I probably didn't really know anybody who did research on ME, and I didn't really know how to get grant funding for research on ME, or who might fund it. So let's go back there. What I did do was have a lot of contact with people with long COVID, and start to do research on long COVID. And this is being approached by a kind of patient activist who some of you may know, called Jez Medinger. And we wrote a book together called the Penguin Long COVID Handbook, which is a few years old now. And that, I suppose, helped, along with all the research papers we were publishing, to bring just thinking about long COVID to a wider audience. And this is the kind of table that many people have published in their publications on long COVID, and it's just the working hypotheses that were on the table from the beginning, of all of the obvious, which I won't go through in great detail now. From the idea of this being a rather cytotoxic virus that is present and can do organ damage in many different organs, or one that's continued to be a very, very strong candidate, which you'll hear more about later. The idea that there are persistent virus reservoirs that are never really cleared, that drive some kind of inflammatory or immune dysregulation. The idea of endogenous herpes viruses, including Epstein-Barr virus, getting reactivated, just general immune perturbation and dysregulation, or the idea of some kind of coagulopathy and endothelial disease or mast cell activation, or autoimmunity, which we'll talk about more in a moment. Because after all of these are diseases where autoimmunity has been shown and have that absolutely hallmark feature of being heavily skewed to females instead of males, in the way that virtually all autoimmune diseases are, or the idea of microbiota dysbiosis, about which more later. So we were one of the labs that was funded to put together cohort studies on long COVID, and the details of this don't matter, but we put together a study called Wilco, which is called Working Out the Immunology of Long COVID. And as shown at the top there, people come in to Hammersmith Hospital and give longitudinal blood samples. And then we analyze a ton of stuff in them, from their symptom profiles and symptomatology to different aspects of their antibody and T-cell immunity and infection history, and many, many different parameters of essentially their immune response and immune function. But one of the things I really wanted to start thinking about and focusing on here in the discussion of mechanism and driver mechanism and common features between long COVID and ME is that certainly a common feature to both of these that we think about a lot is autoimmunity. So to go back to the question of COVID and long COVID for a moment, to me, one of the most striking facts there is that if you think about the very kind of clean data set that we have in the world from that first year of COVID, when the world looked very different. Yeah, we didn't really have vaccines, and the world was divided up into two kinds of people: the people who'd met this virus and therefore, explicitly had seen it, often had been quite ill with it, often had a PCR diagnosis or a lateral flow device diagnosis, and the people that hadn't. And therefore, using electronic healthcare record data sets, one can compare those two kinds of people and say what became of them in subsequent years, and what was the risk, therefore, the relative risk of developing an autoimmune diagnosis in the years afterwards. And the results in every study in every country where this has been looked at has been really striking, that if you had SARS-CoV-2 on board during that year, your risk of a classic autoimmune diagnosis like arthritis or lupus or vasculitis type 1 diabetes went up 2 to 3-fold. Okay, so there's some kind of link between these kind of viruses and autoimmune repertoires. And it's something that we and many other people in the long COVID field and well, to a large extent in the ME field as well have started to look at. And there are very fancy ways of looking at this in the lab, where you can express many or nearly all of the proteins in the human body and robotically spot them onto glass slides, and then probe them with plasma or serum from different people and ask how many of those spots light up as being recognized by autoimmune antibodies that shouldn't be there, you'd assume, in a healthy person, and ask if that's different between these different conditions. And when you do that kind of thing, you get pictures like this, where there are some people who have long COVID who have these clusters of many different autoimmune responses, antibodies to many different proteins in their body, autoantigens, that presumably shouldn't be there and aren't seen in controls, and something we assume or we hope to do with causality of those symptoms in that disease. So all of this led to lots of interchange of ideas and opinions with long COVID and ME sufferers. So what tended to happen to me was that certainly during those 3 or 4 years, I was on the telly and on the radio and things quite a lot, and that was really fun and really interesting. And there would always be a certain number of people who were quite angry with me, especially I suppose both ME sufferers and long COVID sufferers, that we hadn't done enough, which was certainly true. And I'd certainly done nothing for ME at all because I knew nothing about it. And I haven't, to be fair, I hadn't really had the opportunity to know or do anything about it, because I don't believe that at that stage it would have been very tractable to get funding to do research in this area. That, I think, is changing now, as you'll see. So what happened next was that a bunch of us got asked to organize a really big international meeting in Santa Fe, a Keystone Symposium, which kind of framed these questions in a slightly different way, because instead of now having these sort of very siloed meetings on ME or on long COVID, here we were talking about long COVID and other post-acute infection syndromes, and we were inviting all of the people we could think of who worked on long COVID and ME and Lyme disease and post-Ebola and post-chikungunya, and getting people together in the same room to try and work out common themes and common mechanisms that might be driving these post-infectious sequelae. And if that sounds kind of sort of platitudinous, I think it's more than that. As you'll see in the next few slides, the details, I think, are kind of meaningful and helpful, and I'll talk about some of them. So one of the things we end up talking about in those kind of meetings, obviously all over long COVID and ME research, is that we're really held back by our inability to achieve consensus on usable, translatable biomarkers, the kind of things that your general doctor could tick off in surgery as a test to order from your health service that could definitively diagnose you as having one of these conditions and could get you into a care pathway and maybe might inform what kind of therapeutics might be useful or could be used as inclusion or exclusion criteria or study endpoints for clinical trials. And slightly shamefully in the field, we just haven't managed to knock heads together to get that done. So there was a lot of convergence around discussion of that and how to address it. There was also a lot of convergence around, I suppose, central themes that have kind of risen to the fore in terms of those mechanisms I showed at the beginning. The idea of either persistent reservoirs of virus as drivers of disease and or of induction of new autoimmunity as a driver of disease. So if I think about the outcome of all of that kind of new activity and that sort of catalysis of new activity, I mean, I tend to be quite a kind of optimistic person, but I do think there's cause for optimism. I see this as a kind of tipping point where there are a lot of new ideas out there, and there's a lot of new momentum around sort of kicking off new clinical trials, with a lot of cross-fertilization between the different conditions, sort of bidirectionally between ME and long COVID. So this is an example of a trial that a lot of people like out of Norway, with anti-CD38 as a therapeutic that might look rather promising for ME. And I think those kind of approaches will have a lot more traction. So that brought us to this Rosetta Stone proposal. So after many of these discussions and a lot of engagement now with the long COVID and ME communities, just the idea, quite obviously, that there are commonalities between these conditions as rather specific examples of post-infectious sequelae, And surely there are lessons to be learned from this interchange of ideas between the two conditions. So I said, I think it goes beyond the kind of platitudinous down to quite specific details. So what I was doing was that I was reading every possible paper I could over that period, probably many thousands of papers. And it struck me that quite often we were kind of reinventing wheels, and that themes that had come up in ME were being rediscovered in long COVID, and vice versa. So this is one very, very specific example. So I was reading some papers that I really loved about protein biomarkers in long COVID. And so people who like reading the literature, these are all papers that I really recommend. So the one on the left there is a kind of mouse and human paper on kind of central nervous system features in COVID, and says that one key mediator of that might be that's implicated in the studies is this cytokine, this blood biomarker, CCL11. And CCL11 has a lot of interesting properties. It mobilizes particular white blood cells, but it's also implicated in cognitive performance and aging and neurogenesis. It's got immune and neurological functions. And so it looks really exciting in the immunology of SARS-CoV-2. It also comes up as a really big hit in sort of big studies of ME from big pucker groups like Mark Davis's group in Stanford, and it also comes up in the work from Danilo Buonsenso in Rome on pediatric long COVID. So this is us just discovering the same thing over and over again. So so why can't we just get better at acting on it? So stuff like that, or think for a moment about gut microbiota. So everybody knows about the gut microbiota. Folks like us really love it. The idea that many disease processes can be mapped back to the species in your gut microbiome. People have often heard the idea that at a kind of nucleic acid level, we carry around more microbial genes than we do human genes. So we're essentially just taxis for a load of bacteria to carry them around the planet, and they're constantly interacting with all of our body systems, and kind of setting our kind of health set point, interacting with many different parts of our body, including very, very much our immune system. So gut microbiota dysbiosis, the dysregulation of that, is endlessly debated in diseases like long COVID and ME. And if you trawl through the details of that data, the same players come up again and again in the two conditions. So you have this bacterial species called Prausnitzii, where reduction in Prausnitzii is very much associated with ME symptoms, and there's a kind of mechanistic basis for that in terms of the ability of that species to metabolize butyrate and the impact on that on regulatory immune subsets. And if you look at long COVID, exactly the same observations come up. So it doesn't take a genius to rationalize all that into a kind of a framework that can be investigated for how these microbiome differences might lead to kind of sort of symptom changes in these diseases. So sorry, this is a very complicated graphic, but this is the graphic that went into the grant applications for Rosetta Stone, where all we said we'd do is take a sufficiently powered study of, I think, 250 people per group, and just interrogate all of those candidate ideas that have come up recurrently in the literature, from immune analysis to microbiota analysis to oral swabs to look at human herpes virus activation, and try and nail this down once and for all. And so we wrote it, and we sent it to the Medical Research Council, who had a call out for this kind of stuff. And they looked at it, as they do, and then declined it, but said if you tweak it and rewrite it in line with the reviewer's comments and send it back to us, then we'll definitely want to reconsider it. So we did that, and a year later, they redeclined it. So we tweaked it a bit more and expanded it a bit more and sent it to the Wellcome Trust. And they interviewed us about it and liked it, but declined it and said, but if you revise it, we'll have a look at it again. So we spent another year revising it. And then they declined it again. So by this time, 4 years of my life had gone by, and I was getting very gray, and I was almost ready to put the idea to bed. And then I got a very kind approach from the ME Association, who said they'd heard about this Rosetta Stone idea and they might be interested in having a look at it and subjecting it to peer review and seeing if they might want to use the money that really heroic people pull together from all of their marathons and sponsored runs and sponsored activities and contribute to this research, which amazingly they did. And I'm sort of utterly grateful to them and to all the people who supported the ME and the long COVID charities. And now we're at the stage where we're back to recruiting, and people come to this building here, which is the Translational Experimental Medicine part of our hospital at Hammersmith in West London. And they meet our lovely research nurse, Louise, and they do a ton of questionnaires on using the sort of Eleros app, which some of you may know. And they agree to give a blood sample, which they don't find too scary, and an oral swab to look at human herpesviruses, which they don't find too scary. And we send them away with a little stool collection set to give a tiny collection of poop, which they do for some reason find much scarier than giving away blood. But honestly, it isn't very scary and really, really helpful for assessing those microbiota differences. So we're just hoping in that way that with a bespoke study to interrogate all of these clues that are out there in a really properly powered side-by-side 21st century way, we can come up with some answers that really illuminate mechanism and causality and truly offer you some kind of Rosetta Stone decoding. So that's what they did. That's what it is. So it's out there. We're very much recruiting. We hope it's going to make a difference. So all I can say is that's how we've come at it. Thank you to everybody who supported it. And if you're interested in what we're doing or interested in taking part, then get in touch with me. Thank you. So I'm going to stop sharing.
Many thanks, Danny. That was phenomenally interesting. And we wish you all the best of luck with the investigations going forward. I've been monitoring the chat and the Q&A button. I think you've just answered one of the questions, which is, can we still sign up for the Imperial College study, which I think is a yes. Correct. And
can I annotate one point there that you'll have seen in the Rosetta Stone thingy that we talk about healthy controls. That means people who who've had SARS-CoV-2 and got better, or people who don't have and have never had ME. You'd imagine that there must be billions of people like that in the world. They're the people that we find really, really hard to recruit. So if anybody has friends who are healthy and don't have any of these conditions, please bring them along.
That's a very good point. I think we should disseminate that as far and wide as we can. I have one question, and then I'll follow up with another question, which is a little tangential, but maybe you have some insights. So my question would be, with the autoantibody work, you're seeing huge numbers of autoantibodies in long COVID, very few in the controls. To what extent do you think that just reflects a sort of the aftermath of a condition that causes severe tissue damage, rather than a specific signature linked to long COVID?
The first thing to say is obviously these are signals that go years out. So these are people who are being looked at 3 and 4 and 5 years after their SARS-CoV-2. So so it's a little bit special and a little bit weird, but the point stands in the sense that we've got new and better everybody has new and better technologies for looking at autoimmunity far more comprehensively than medical research has ever been able to before. And I guess the time is now to uncover how much autoimmunity is out there. But certainly it's true that viruses driving these diseases, and that's probably true of things like EBV and flu in ME and of SARS-CoV-2 in long COVID, can turn on new B cells to make new autoantibodies, which I'm guessing is something to do with the underpinnings of disease. But let's have a look and see.
Great. One comment from Selena Shaw and Action Fremy. Could you include the email address in the chat for people who want to join your study? that would be great. And then there was an earlier question from Ulrika, which was asking about whether there are or what the best treatment options or therapies might be for mitochondrial diseases, and whether that might have an autoimmune basis, if you have any insights on that.
I think it could easily have an autoimmune basis. I'm struggling to think if any of the the sort of autoimmune toolkit would specifically address that. There might be some other aficionados on the call who can answer that better than I can, because I can't think of any of the treatments that have been proposed that would specifically get at that.
Yeah,
I'm going to type in the address in the chat.
Thank you. And final question that I think we can take before we move on. Could EBV be important in creating autoantibodies years and decades later?
Yeah, I mean, we could talk about that for days, couldn't we? So the fact that EBV is a sort of puzzling virus, isn't it? Because nearly all of us have it, and yet it is so massively implicated in different autoimmune diseases, including things like lupus and multiple sclerosis. And and why shouldn't it be? It can turn on B cells, it can live in B cells, it can activate plasmablasts. Absolutely, yes, we've got to get to the bottom of it.
Fantastic. Thank you very much, Danny. I think that wraps up the questions. So we'll move on to our next speaker, who is Andy Malinovsky from Precision Life. I've not met Andy personally, but reading his resume, I think it's very clear that he's far more intelligent than I am. He's held many positions across Europe. His work now is focused on essentially using scientific data to generate drug discovery pipelines, particularly genetic data, using AI, and he's here to tell us about those efforts in both long COVID and ME/CFS. Thanks, Andy.
Thank you very much for the kind introduction, David. I just want to make sure that everyone can see my slides.
Yes, we can. Well, I can. Yeah.
Amazing. Yes. So yes, it's a great privilege to be here and being able to present on the behalf of Precision Life. Also, big thanks to Danny for a great introduction to both diseases, meaning that I can jump straight into my topic, which is genetics. So essentially, I'll be telling you about how we've been using our combinatorial approach to better understand genetic similarities between ME/CFS and long COVID. And I will try to cover 3 parts. So first of all, I will set the scene with our initial study of ME, and I will take this opportunity to also introduce our combinatorial approach, our methodology. In the main part, I will talk you through 3 different approaches for exploring commonalities between ME and long COVID based on results of our more recent studies, and I will finish up with conclusions and some potential clinical applications of our findings. So in our first ME study, we performed hypothesis-free combinatorial analysis of the UK biobank cohort. So why is it called combinatorial? Because unlike GWAS-based methods that tend to look at single SNPs, so single nucleotide polymorphisms, we look at combinations of these SNPs that are together strongly associated with disease. And we call them disease signatures. So importantly, they capture both linear and nonlinear interactions between genetic factors. And what this allows us to do, it allows us to do 2 things. So by clustering these disease signatures by patients in which they co-occur, we can stratify the disease into different subgroups, which is shown here by these clusters of different colors. And then by mapping these SNPs to genes and then further to pathways, we can start understanding potential mechanisms driving disease in these specific clusters. So I'll give you an example. This green cluster on the left-hand side. So the key SNPs we identified in it is in a gene called ACAP1. This gene codes for a mitochondrial scaffold protein. and this is involved in replenishing ATP supply after exertion. So interestingly, what we can then do, we can check if the symptoms of patients in this specific green cluster are consistent with biological mechanisms around ACAP1. And indeed, patients within that subgroup report fatigue and post-exertional malaise as their main symptoms. So similarly, we found other subgroups and candidate genes with distinct potential mechanisms, which included processes like infection response, neurotransmitter biology, circadian rhythms, and for all of these, we were able to find matching clinical symptoms within these subgroups. And I think this is important because this is consistent with what the others have shown, basically demonstrating, well, suggesting that complex diseases such as ME and long COVID have several different disease drivers. So this analysis that I just briefly outlined to you was completed in 2022, so a few years back. But then we were able to confirm and expand our findings in further studies, all of which have been published. So first, in our primary study of long COVID, we used the SANO Gold patient dataset, and we identified long COVID-associated genes. This was followed by a reproducibility study where we confirmed the long COVID gene associations derived from the SANO study using an independent cohort, which was the All of Us patient dataset. And finally, the study I'll be mainly focusing on today called LOCOMI, which was basically just accepted for publication. I think it was last week. So in this study, we analyzed the DECODE ME cohort, which many of you will be familiar with. And with this analysis, we identified over 250 candidate core ME genes. And more relevant for this particular topic, we also found that a significant number of long COVID genes that we identified in our previous studies are also associated with ME, and I will go back to this overlap in a minute. So if I can go to the next, oh yes. Right. So to study genetic commonalities between ME and long COVID, we have used 3 independent but complementary approaches. And in each approach, we evaluated overlap between different gene sets. So I'm just going to explain it in detail now. So in our primary approach, we looked at overlap between reproducible ME genes from the DECODE ME analysis, and compared them with reproducible long COVID genes identified through looking at the SANO Gold. Our primary long COVID analysis, but these genes were also reproduced in all of us. In our second approach, we compared top-ranked ME genes that were derived using an independent machine learning model with long COVID genes from the SANO analysis, so essentially similarly to what we did in the first approach. And finally, in a more exploratory analysis, we assessed overlap between genes from DECODE ME and a set of genes which were reported to be differentially expressed in long COVID patients, so essentially leveraging data from a published study. And now I'm going to talk about these 3 approaches in more detail. So the primary overlap analysis between ME and long COVID genes were done, as I already mentioned, based on DECODE ME and SANO results. And we found that 76 out of 180 long COVID genes that we previously found to be associated with ME were also, which were previously found to be associated with long COVID. We found them to also be associated with ME. And this represented a statistically significant finding. And then importantly, we saw this significant overlap also after controlling for gene length using a length-matched permutation test, which is something you can see in this graph. So essentially, the red diamonds shown here, they show observed number of overlapping genes. And you can appreciate that these values are significantly higher than the numbers represented by box plots. And these box plots, what they represent, they actually show what the overlap would be, what the expected overlap would be if we randomly selected the same number of genes. And the most important point here is that this enrichment is true across 3 different gene lengths categories, which are shown on the x-axis. So starting from short genes to mid-length genes, and then to longer genes. So what we did next, we took those overlapping genes that we identified in the slide, in the analysis shown on the previous slide, and we performed the pathway enrichment analysis, first using the gene ontology biological processes terms. So we identified a number of relevant pathways which fell under 4 broader biological processes, which you can see in these boxes here. So synaptic biology and neurotransmission, CNS development, cardiovascular biology, specifically around smooth muscle function, and finally, mechanisms related to cell-cell contact. So to check if this enrichment actually reflected context-specific mechanisms related to the diseases rather than generic pathways, because finding these general pathways is something that can happen if one is using pathway enrichment tools. So we went back to the underlying genes and verified that they were expressed in relevant tissue cells. For instance, genes that were highlighted as a part of the synapse biology term, such as neuroligin 1 and synapsin 3, are indeed enriched in the brain and specific neuronal populations. And finally, we observed similar pathways coming up when we used another database called Reactome. In our second approach, we established an independent machine learning model for ranking ME genes, and this was trained on the DECODE-ME data. And next, genes associated with 3 different long COVID phenotypes that came from the SANO cohort were scored by their SNP ranks within this model. So essentially, when you look at the y-axis, the higher the position on the y-axis, the higher the SNP rank and higher importance. So this analysis, essentially shown that SNPs mapping to long COVID genes were significantly enriched in top-ranked ME SNPs, and this is something that is highlighted by these ellipses here. And this was not the case for randomly selected genes. So all genes on the left-hand side, which you used as a control. And this result was especially clear for gene sets that were identified in severe and fatigue dominant patient cohorts, where we saw that 25% of long COVID SNPs ranked in the top 0.5% and 2% of ME SNPs respectively. So in other words, this model notices many of the same genetic signals that have been already associated with long COVID and does it in the context of ME. Okay. And sorry, one thing I forgot to mention, and which is important, there are also plenty of long COVID SNPs that are lowly ranked in this model, which you can see towards the bottom of the y-axis. And we think that these might potentially represent non-shared genetics and non-shared biology between the diseases, which is actually something that we will be looking more into in the future. Next, we wanted to look at some top priority overlapping genes coming from these analyses. So essentially, we considered long COVID genes that were overlapping with the highest ranked ME genes identified from the machine learning model. And as a result, we obtained 3 top-scoring candidates. So there was MAPK9, a protein kinase linking inflammation, oxidative stress, and metabolism. And this came with previous evidence linking it to both diseases. Second one was neuroligin 1, which is a neuronal protein that modulates glutamate transmission, and genetic variants in this gene, as well as altered expression of this gene, have been linked to ME. And finally, interesting thing is that this gene is regulated by clock genes, which we previously identified as a part of the circadian rhythm mechanism in the disease. And finally, PT-PRD, this is a transmembrane phosphatase, and this regulates neuronal adhesion and cognitive functions. Again, genetic variants in this gene have been linked to ME in previous studies, and also linked to other conditions involving brain inflammation, making it relevant. So as you can see, top-scoring genes had previous links to one of both diseases and are implicated in disease-relevant biological mechanisms. And finally, an interesting finding is that all these 3 genes were found in the fatigue-dominant long COVID cohort. So this might suggest that ME might be genetically overlapping specifically with the fatigue-dominant subtype in particular. And our third approach, which I said has been a little bit more initial and preliminary. So this was essentially investigating genetic and transcriptomic overlap. So not genetic, but genetic transcriptomic. And we used a published set of genes found to be differentially expressed in patients experiencing long COVID symptoms. And then we compared this set against the candidate cord ME genes that we identified from Decode-ME, and as a result found 22 overlapping genes. So it's important to say here that the transcriptomics data set from this paper was generated from blood samples. We know it was relevant only to a proportion of genes and mechanisms identified in our analysis, and we expect the number of overlapping genes to go up once there is data from additional tissues. Okay, so I will wrap up now. So to summarize, our genetic results support ME and long COVID being 2 distinct but partially overlapping conditions, which is consistent with previous findings, especially ones around symptomatic overlap and differentiation studies. The genetic similarities we observe, they suggest shared biological mechanisms related to specific subgroups or subtypes in each disease, and these mechanisms can be targeted therapeutically using our stratification insights. So what I didn't tell you about the disease signatures is that we can also use them as patient stratification biomarkers to identify patients who are most likely to benefit from a modulation of specific target or mechanism. So we believe that the fastest and most cost-efficient approach to deliver effective treatments to ME and long COVID patients is precision drug repurposing. And we have previously analyzed, at Precision Life, we analyzed drug discovery pipelines to find on-market and in-development drugs modulating the same targets that we identified, but we identified them in novel indications. And the same is true for, we found the same in the Lokomi study. So we identified several such repurposing opportunities, which basically are modulating the 18 candidate genes shared between ME and long COVID. So this is obviously the proportion of all the genes that we find shared between these diseases. And in the current work, we are prioritizing safe and easily accessible drugs, focusing on generics, and the process is outlined in this graph here. So we will be testing these prioritized candidates in proof of concept small studies using genetically stratified cohorts, which will be selected using our genetic mechanistic patient stratification biomarkers. And the aim here is to demonstrate the disease-modifying potential as well as the right dose and safety. And finally, I would like to thank the whole Precision Life team. This is I'm presenting on behalf of the team. Special thanks to Jason and Shayoni, who have been leading this work, and thank you very much to all of you for your attention. And I'm looking forward to taking some questions. And I think I can stop sharing now.
Yes, please stop sharing, Andy. Many thanks for a fantastic talk. It really is great work, hugely valuable to the community. We have a couple of questions off the bat in the Q&A. One, what someone is asking, will more be done to study the neuroinflammatory gene, which would help change perceptions of the disease?
Yes, well, definitely this is something, this is a great question. This is something we are looking into right now. Neuroinflammation is one of the sort of core mechanisms that we find associated with the disease. Obviously, one of our priorities here is to also find candidates that have matching candidates with a potential of repurposing. But obviously, we are also interested in looking at the more novel disease biology and finding the right druggable targets. So the short answer is yes, we will focus on this.
Great, thank you. We've got two more questions coming in. Someone is asking, are you able to name any of the potentially repurposed medicines that might be valuable in treating ME/CFS?
Yes, so I think the best way, the best thing I can recommend is to encourage you to read our most recent publications. So as I said, this is now should be in press, and of course the preprint is available. So in this publication, I can briefly mention we give two examples. So one is an asset targeting innate immunity receptor TLR3. Having said that, this specific asset, we find strong signal for it, especially in ME, and not so much in non-COVID. So this is not sort of the direct focus of the overlap work. And then the second candidate is apremilast, which is a PDE4B. It is a pan-PDE4 inhibitor, and we find signal for it in both indications. It's not a perfect overlap because, as I said, this targets all PDE4s, and we see a strong signal for PDE4B in ME. And in long COVID, we see a signal for another isoform PDE4D. However, this asset is known to modulate. So basically, the highest affinity targets are exactly these two isoforms of PDE4.
Fantastic, thank you. We've probably got time for two more quick ones. One person's asking, what are the barriers to identifying potentially more risky drugs? Is it funding, regulatory, other things?
I mean, this is a very good question. Well, I can offer some thoughts, initial thoughts. I think central to what we do is patient safety at this point. So that's why we are focusing on generics because they come with established, well understood safety profiles. Obviously, cost is another thing to consider here. Basically, cost of acquiring, of sourcing these drugs and testing them in clinical trials. So I think these are two main reasons why. And obviously another reason is that these two diseases are basically they don't have very well established in vitro and in vivo models, and obviously before testing these drugs in humans, it would be great to, especially thinking about the more sort of less known targets. I would think that this would be sort of the first step, but we are limited here because of the paucity of the models.
Thanks very much, Andy. One last comment. Someone's asking, this is a good comment. Could you post the link to your paper in the chat, the preprint?
Yes, absolutely. Yes, I'm gonna do it right away.
Lovely. Thanks, Andy, for a fabulous talk. Very exciting stuff. We look forward to hearing more down the line.
Thank you.
And now we move on to our third speaker. It's a great pleasure to introduce Amy Proal. Amy is a global microbiology superstar whose research is centered around really the concept that many chronic diseases are driven by covert infections. And I think the implications of that are hugely apparent when we think back to things like the discovery of Helicobacter pylori underlying chronic duodenal ulceration, which has transformed a chronic disease to a highly treatable disease. So this concept is something she pursues globally through her research foundation, which she set up with Mike Ben-Elzaka. They're called PolyBio, if you haven't heard of them. And I know we and many others are hugely grateful to PolyBio for supporting research into long COVID and keeping global momentum going when governments had largely neglected funding for the disease. So great pleasure to introduce Amy Proal. And yeah, Amy, please feel free to share whenever you're ready.
Great, and thank you, David. I look forward to speaking now. Let me show slides. Oh, do you see presented view? One moment. Slideshow. Okay. There. Do you see my first slide?
Yes, I can see it.
Awesome.
Yeah.
Okay. So yes, as David said, I'm Amy Proal. I'm a microbiologist. I have two jobs. I lead and co-lead PolyBio Research Foundation and our long COVID, ME/CFS, and chronic tick-borne vector-borne illness research programs. And I'm also the scientific director of the Cohen Center for Recovery from Complex Chronic Illness, or the CORE, which is a long COVID and related infection-associated chronic condition clinic at Mount Sinai. So today I'll be talking about long COVID and ME/CFS, why persistent pathogens matter. And okay, I think everyone knows this, but when we're talking about long COVID and ME/CFS, there are a couple differentiating factors just in terms of the infections that people sustain. Long COVID, we know it has a SARS-CoV-2 onset. This is obvious.
Sorry, Amy,
Okay.
sorry, someone's just asked if we're looking at your computer screen rather than your slides.
Okay.
Says the screen I can see says BMC infectious diseases.
Okay. One moment. Mike, can you help me share my side? I'm gonna just try to, okay. And to share this PowerPoint.
There we go. I think we
Okay.
just need to press slideshow, and I think
Yeah.
you should.
Here, wait, just one moment. Sorry. Just good. Okay, that's fine. Okay.
That looks good. Yeah. Yep.
Okay, great. Okay.
Thanks. Thanks, Mike.
No problem. Yeah, thanks. That was Mike Ben. I was like, my co-founder. Okay. He's better at slides than me. Okay. Back to the onsets. We have a SARS-CoV-2 onset with long COVID. ME/CFS, we all know, has different onsets. It doesn't even have to be an infection. Sometimes people develop ME/CFS symptoms after an injury, after a traumatic event, after other exposures, but there have been pathogens historically implicated and even implicated in some research papers that are connected to the ME/CFS disease process. Those include the herpes viruses, the enteroviruses, and chronic tick-borne, vector-borne pathogens. So in at least some of these cases, a consideration is that in long COVID and ME/CFS, the pathogens that initiate or exacerbate symptoms may persist in patient tissue or host cells. And our group, including David, that is called the Long COVID Research Consortium at PolyBio. We wrote, for example, this paper in 2023 on SARS-CoV-2 reservoir in post-acute sequelae of COVID, or PASC, which is another word for long COVID. And what we do is we review research, even at that point in time in 2023, showing that the SARS-CoV-2 virus can persist after acute illness, especially in tissue, RNA or antigen in different cases. And in some cases has been found in the tissue of patients with long COVID symptoms. This is a more recent paper by a group that actually goes into different targeting treatment strategies for persistent SARS-CoV-2 in patients with long COVID, and also has a review at the start on different studies that have found persistent RNA or antigen in long COVID patients. But one example of that is this study that was published by Michael Peluso and Steve Deeks and their group at UCSF. They do some really cool imaging showing T cell activation throughout the brains and different regions of long COVID patients. But also you can see there, that is a gut tissue sample on the right. It shows the pink is double-stranded spike RNA. This is collected from a long COVID patient. This is almost 2 years. In the case of, the second image, it's 676 days post-COVID. The other is 442 days post-COVID in their gut tissue. And double-stranded RNA suggests the virus was replicating at the time that this tissue sample was analyzed, so it isn't necessarily inert. And this group, which is exciting, will be publishing a preprint with many more analyses of gut tissue and actually other tissue samples collected from long COVID patients in the next weeks. And overall, they find persistent RNA and antigen, including more double-stranded RNA in a little over 30, up to 40% of people with a long COVID diagnosis. And you do have to consider the fact that this is a very small piece of tissue. So, there could be patients with long COVID who have persistent SARS-CoV-2 RNA or antigen in, just a piece of side tissue that wasn't collected via the procedure to get the gut tissue. So 30% is actually pretty high. And the key thing is in the work that you'll see them preprint soon, they use spatial transcriptomics methods to characterize the immune environment and gene expression changes near identified RNA. And there's definitely upregulation and downregulation in the immune system near identified persistent RNA, including actually some pretty important downregulation of the immune response, including natural killer cell activity. So the virus does seem to be doing something, and more research is needed. And we don't necessarily think that every patient with long COVID has persistence of SARS-CoV-2. We think it's likely a subset, but it's still important. And then when we go to ME/CFS, when we were first at PolyBio, creating a program that was helping people to study SARS-CoV-2 persistence in long COVID, the reason that we did that early on was because of research done in ME/CFS before COVID started. This is a paper by John Chia, who's at the Enterovirus Research Center in California in the US. And this is a paper he published all the way back in 2008, where he got gut tissue, almost similar tissue to what we've been collecting on patients with long COVID, and use staining to look for protein created by an enterovirus. And enteroviruses and found it. So there are a good portion of participants in this study who were positive for the VP1 capsid protein of enterovirus in their gut tissue. And to better describe the enteroviruses, what's really interesting is they have similarities to SARS-CoV-2. They, like SARS-CoV-2, are single-stranded RNA viruses. They are acquired via respiratory infection often, and they often reach the gut. Disease can be totally mild, asymptomatic in some cases, like SARS-CoV-2, or extremely severe. And they're not studied too often for their chronic capacity, but they are capable of persistence. In fact, persistence of enteroviruses in diabetes in the pancreas is a big area of research, actually. And non-polio enteroviruses cause about 10 to 15 million infections and tens of thousands of hospitalizations every year in the US alone. So these viruses are out there infecting people a lot. So, when you see this work of, persistent enterovirus RNA in ME/CFS, it is of great interest. And, going back again, because some of the best work on enteroviruses that we're working now to replicate with more modern methods, this is a paper from 1994, and it's one of the autopsy studies that was done on a patient diagnosed with ME/CFS, or chronic fatigue syndrome. And in this case, they identified enteroviral RNA in the heart, muscles, hypothalamus, and even brainstem of the patient. It could be that enterovirus can even get into the central nervous system in some of the patients who have ME. And, more research needed, but this is interesting. And, when we think about ME and the myalgic encephalomyelitis diagnosis, the way it's named is myalgic means muscle pain, encephalo, brain, myel, spinal cord, itis, inflammation, right? So basically, muscle pain associated with swelling of the brain and spinal cord. And, it's important to understand that the enteroviruses are viruses that drive those kinds of symptoms. In fact, when, the term benign myalgic encephalomyelitis, the name we have derived for ME, is actually coined in this 1956 paper in The Lancet. And this was during a time when there were different outbreaks across different geographical regions of cases and clusters of patients who were diagnosed with what became known as ME. And you can see that on the right, Coxsackie viruses or poliomyelitis was tagged and was identified in some of these epidemics, despite the methods that they had to identify what viruses or what infections were, impacting people being not that great at the time. And so the reason that they named it benign myalgic encephalitis was in classic polio. As we know, there was, paralysis in many cases. And what they were seeing in these patients was a polio type illness that did not result in, paralysis. And for that reason, they said it was benign myalgic encephalomyelitis, but it still had this poliovirus-like presentation. So, the study of chronic enterovirus persistence really goes back to the roots of the ME diagnosis, but has been really understudied in the field. And so we're really glad that we were able to get a, really generous donor to support the group at UCSF, who I showed you the data on the SARS-CoV-2 virus in gut tissue from long COVID patients. They're now collecting gut tissue from patients with, a ME/CFS diagnosis prior to COVID. They're using advanced methods to look for enterovirus RNA protein and many other immune and other parameters in gut tissue and other samples from these patients. And I think the data will be really, hopefully, interesting. And so I think then this brings me to, the connections between long COVID and ME/CFS. If you're looking at these trends where we think that maybe some patients, at least some people with this diagnosis don't fully clear the pathogens that they get, and there can be persistence of those pathogens that could drive disease, it's just really important to understand that there's redundancy in the way that different persistent pathogens can drive disease. They can do similar things. So if you look at the central symptoms of ME/CFS and often long COVID, we all know them, the flu-like symptoms, the malaise, the post-exertional malaise, the autonomic dysfunction, the pain, the nausea, the cognitive dysfunction, the headaches. All those symptoms scream, and this is where my colleague Mike Van Elseker has been awesome because he's a neuroscientist at Mass General. Brainstem, at least, if you think about those symptoms, the brainstem is where the vagus nerve, a very important nerve that innervates most trunk organs of the body, sort of innervates, the gut, the heart, all of these important organs. It enters the brain at the back of the brain at the brainstem. And nuclei that actually even overlap, you can see in that figure that Mike and I published in a paper together. There are nuclei overlapping in that region of the brainstem that control what's called the sickness behavior response. So that's literally when you feel, if you get an infection and you start to feel sick, like you want to lay down, like you don't feel well, the malaise, that's because the body is mounting these sickness symptoms by the vagus nerve sensing the infection in your body, signaling the brainstem. And that region of the brainstem, the nucleus of the solitary tract, creating these sickness behavior symptoms to essentially let you know you're sick. There's also nuclei in the brainstem that control pain, nausea, and then autonomic functions like heart rate or breathing. So this area of the brainstem really is connected to a lot of core symptom signaling associated with ME/CFS symptoms. So here's the thing, look, if the vagus nerve, senses inflammation in other parts of the body or in the brain itself, it will convey a signal to the brainstem, and those sickness, pain, nausea, autonomic symptoms can result. And so here's an example of how this could happen if someone has persistent SARS-CoV-2 in the gut, right? The virus there in the gut could lead to inflammation sensed by the vagus nerve, signal conveyed to the dorsal brainstem, resulting in sickness, pain, nausea, autonomic, and other symptoms. And this person could meet the criteria for ME/CFS. And, honestly, this is a really interesting study. We also sometimes think that the vagus nerve itself could be directly infected by pathogens that persist in people. This was an autopsy study done on people with acute COVID, but they did find that in every single body analyzed where they looked at vagus nerve, SARS-CoV-2 was actually in the vagus nerve itself. So these pathogens could literally infect the vagus nerve, which would be the most sort of direct way to cause these core ME/CFS symptoms to happen. But here's an example where a person who was sick 20 years ago with an enterovirus infection could have persistent enterovirus in their gut. Again, the vagus nerve would sense the inflammation associated with the infection, convey it to dorsal brainstem, results in sickness, pain, autonomic symptoms, and this person could also meet the criteria for ME/CFS. Now, if you look at here, other factors could converge to also lead to similar symptoms diagnosed as ME/CFS. So for someone else, another patient could have microbiome dysbiosis or imbalance of the gut microbiome, leading to inflammation in the gut, again, sensed by the vagus nerve. And in their case, they may have a structural issue also, like craniocervical instability, in which the ligaments in the back of the neck are actually not in good shape and the head sort of sinks down and compresses the brainstem. Those two factors could converge in this patient to lead to sickness, pain, autonomic symptoms, derived from brainstem signaling. They could also meet ME/CFS criteria. And I can go on like this. There are patients who could have missed the fact that they were bitten by a tick at some point, which is something we see more often, or even were bitten by a tick and just were not adequately treated with antibiotics at the time of a tick bite. And Borrelia, the Lyme pathogen, could still be in their symptom system, and there could be a same way that pathogen could really drive some of the same, signaling through vagus nerve, brainstem, and other related pathways. Again, diagnosed with ME/CFS. Literally, here's just another example. Babesia is a parasite that can drive similar symptoms. It's another player, right? So this is the thing is, what is possible in patients with an ME/CFS diagnosis is that people accumulate different infections, but there are common ways in which those different infections can result in chronic symptoms, and people can be diagnosed with ME/CFS. And as you guys probably know, anyone who's a physician knows, many people who get diagnosed with ME/CFS have what we call multiple hits, where they will, one infection started their symptoms, but another infection or exposure exacerbates their symptoms. And so there can actually probably be ways in which maybe multiple pathogens that are in the person can converge to potentially drive symptoms also associated with ME/CFS. And as you know, the injuries, the other insults, the trauma can sort of add into these hits in a way that can make the symptoms manifest the most. And talking about Babesia, I just want to say that Babesia is a parasite. So I think what's important to understand is that chronic tick-borne vector-borne illness is pretty undiagnosed across most of the US and definitely in parts of Europe. And ticks carry not just Borrelia, the Lyme pathogen, but also many other organisms. They carry Babesia, which is a parasite. They carry Bartonella, which is an intracellular pathogen that can drive many chronic symptoms. And Babesia drives really classic symptoms of ME/CFS. So it drives fatigue, difficulty exercising, difficulty with sleep. Those are all key chronic Babesia symptoms. This is just an interesting study about Babesia, which showed that low haptoglobin is a common finding in babesiosis, indicating problems with the red blood cells. And this is just a paper that I thought was interesting where in this paper, ME patients demonstrated reduction in haptoglobin levels following post-exertional stress, and lower baseline haptoglobin concentrations were associated with impaired cognitive performance. I don't know, but when I look at these papers, I'm like, interesting. Is there some relationship there between then some of these patients possibly being exposed or having a pathogen like Babesia? And right now at our clinic at Mount Sinai, we are running the first clinical trial of tafenoquine. It's a treatment that can target Babesia as a persistent pathogen. And at our clinic, we see patients with a ME/CFS and long COVID diagnosis, and there are patients from both those patient populations who test positive for Babesia, and we put into our trial. So in other words, we are currently running a clinical trial of tafenoquine for chronic Babesia that has patients with an ME/CFS and long COVID diagnosis in the trial. And last, one of the most explored and ignored organisms transmitted by not just ticks, but also by other vectors, including just animals. So example, just dogs and cats can carry fleas that carry this is the organism Bartonella. There are many ways to get Bartonella simply from just exposure to other animals in addition to ticks. Bartonella in a chronic form can drive, some of the top symptoms associated with ME/CFS diagnosis: myalgia, muscle pain, bone pain, a lot of symptoms that patients with ME/CFS can suffer from. And, this is a case of a post-COVID patient, but we see a good amount of Bartonella reactivation after COVID. This is likely because Bartonella that's latent infects the vasculature or the red blood vessels. And the SARS-CoV-2 spike protein targets the blood vessels in some way, which can probably be a pattern allowing previously dormant or latent Bartonella to potentially manifest and drive more symptoms in people after COVID. So we are, doing quite a few more studies now in patients who have both long COVID and ME/CFS diagnosis to look for Bartonella, and there are many different strains of Bartonella in their samples to make sure that we're accounting for the persistence or possible contributions of that pathogen in people with an ME/CFS diagnosis. So overall, this is a tweet I wrote. If you're a patient that meets ME/CFS and post-exertional malaise criteria, the information on the specific infections or exposures that led to the onset or exacerbation of your symptoms are of major importance. This can help you pursue personalized treatment. That's what we see. What we see is that patients come to us with a diagnosis of ME/CFS, and we don't assume that everyone has the same thing. We think that the infections and exposures that they have are key to their case. We think that some of the infections could still be in their bodies. And if we're able to break people up into, the testing for those infections, we're sometimes able to treat people in a way that can really improve their symptoms. For example, we've had patients with an ME/CFS diagnosis who have tested positive for Lyme disease via even CDC criteria, and we've been able to put them on extended courses of antibiotics or other drugs and gotten them to a place where they are greatly improved or even better. So I know that this is sometimes a different view of ME/CFS than other groups take, but we really do think that these infections and kind of the common mechanisms by which they can drive symptoms are key to understanding the best way to study and to treat patients with these diagnoses. A key consideration is that the diagnostic test that can help doctors identify if a person diagnosed with ME/CFS or long COVID has these infections are not that great. So right now, even there is no commercially available clinical test for persistent SARS-CoV-2 or spike protein. We're working on that. We actually have a PolyBio program that's currently taking some of the top assays and working to validate and commercialize them so that a doctor could actually better measure persistent SARS-CoV-2 or spike protein in a long COVID patient, but that doesn't even exist in the medical world right now. And really the same thing goes for some of these other pathogens. The main forms of testing that you can do are, antibody based. And a lot of times those are not just, that great, because one of the paradoxes with some of these infections is that the longer that the person has the infection in their body, the more it downregulates parts of the immune response and the more the patient actually has problems correctly mounting antibody responses, IgG or IgM, to its presence, meaning that some of the sickest patients can paradoxically test negative via antibody testing for pathogens that they might have. So key to this is that we really need, a next generation of diagnostic tests to better identify these different infections in people with an ME/CFS and long COVID diagnosis. And we're working on that. A huge part of the tests that need to be developed is that, the persistence, and you can see from some of the examples of these pathogens, is often in the tissue of patients. So in the tissue of their gut or the tissue of their brain, and not in their blood. So an easy blood test to try to find it is not going to work. It will make the person look negative, that they don't have the infection if you just look in blood, when they could have the infection throughout tissue from the body and brain. And so the diagnostics that we're working to innovate are diagnostics, for example, that capture extracellular vesicles or small vesicles derived from tissue that still have viral antigen or other, associated with inflammatory markers, so that we can capture those in blood. In other words, they're diagnostics that are able to better capture tissue-associated signals of pathogen persistence in more accessible fluids. It's a huge focus of what PolyBio is doing right now, with the goal that we can create this next generation of diagnostics that will better allow the study of these pathogens in ME/CFS cases to be as accurate and innovative as possible. So I'll stop there.
Thanks so much, Amy. That was a real tour de force, a fantastic talk. We have a little bit of time, and we have a few questions, quite a few questions. I'm not sure we'll get through all of them, but so number one, I'm going to concatenate these questions. One person is describing persistently high spike protein in the blood for many years, and another person is describing persistently high antibodies to spike in the blood for many years. The questions are, could these reflect ongoing viral replication, and at what point would you expect your spike levels and your antibody levels to drop off if you did not have persistent infection with SARS-CoV-2?
Right. That's a good question. We need a lot more research on it right now, but I think one of the things that is a trend that we're looking at in long COVID is that, let's say the virus itself is persisting in the tissue and the lining of the gut. It's possible that during periods it does translate spike protein that leaks into the blood, where the protein can be measured. And so there are a couple teams, David Waltz at Harvard, several groups now who are, creating sensitive tests that can find small amounts of the spike protein in blood from long COVID patients. And give or take a percentage, maybe 20, 30% of long COVID patients in our various studies are positive for spike protein. We do think that that is likely derived from tissue-based reservoirs of the virus itself. In other words, we don't think it's really the spike protein persisting alone. We do think it's likely derived from virus. And so I think that if you, have spike protein in your blood and by a test that's reliable, you would want to, and again, we need to innovate this, and we're working on, some of the first clinical trials in the space. For example, Invivyd is a monoclonal antibody company that we've been working, we formed a group, me, Akiko Osaki, Michael Peluso, David Petrino, with their company to start one of the first clinical trials of monoclonal antibody combined with an antiviral for the possible treatment of persistent SARS-CoV-2 and long COVID. And I hope that that trial is announced soon. But, right now, the treatment options out there wouldn't be perfect for you, which we need to get on that. But I would definitely think that if you have spike protein in your blood, you would want to consider that you haven't cleared the virus.
Great. Related question, for those of us with long COVID who can't access antivirals, are there any supplements with antiviral activity against COVID that show any promise?
I know, and we know that it's a difficult time now in terms of antiviral access and the treatments needed for possible persistence of SARS-CoV-2. I think that there's two approaches in trying to target a persistent infection. One is to use antivirals or other drugs to try to knock down the infection itself, and one is to actually support components of the immune system to allow the immune system to maybe better target the persistent virus. We have a clinical trial at Sinai of low-dose rapamycin where we're actually testing that hypothesis, and we have not fully finished the trial yet. But in studies of healthy older adults, clinical trials, a once-a-week dose of rapamycin was actually shown to activate parts of the immune system. So at the end of taking a low dose of rapamycin, the healthy older adults had increased interferon signaling in one study. They had some improvements in T cell exhaustion. And interestingly, when they were given the influenza vaccine at the end of the trial period, they had a more robust response to the vaccine. So, this is preliminary, but one of the reasons we're running that low-dose rapamycin trial at Sinai and Akiko Osaki's measuring these immune and interferon parameters is because we maybe think that drug taken in a dose could support the immune response to better clear the virus. But again, this is so early. I don't have recommendations that I can just give that are concrete or that have huge evidence base behind them. Just that some of those are the things we're trialing.
Great. Thanks, Amy. A couple of other questions on other viruses that are reactivating and our tests or suggestions again for supplements to tackle reactivated EBV, HHV6, etc.
Yeah, and that another area where HHV6 especially has been associated with ME/CFS cases over the years, and I think that it's an underexplored virus and contributions to the general sort of chronic fatiguing symptoms that people get. I actually got to know Dara Mablashi, who discovered the HHV6 virus. He was a friend of mine until he passed away a few years ago. He really thought that virus should be better studied in ME/CFS, and we're trying to do that. But in the meantime, again, with the herpes viruses, there's a protocol that we are again almost starting a trial of at Mount Sinai that combines valacyclovir with Celebrex. Celebrex is a COX-2 inhibitor. The herpes viruses sometimes have been shown to upregulate COX-2 signaling, so a COX-2 inhibitor could potentially address their activity. So we are going to trial it along with valacyclovir in patients starting with long COVID who have evidence of herpes virus reactivation. But again, these are just, hypothesized combinations of antivirals at this stage, and we need the clinical trial data to tell us what, really works.
Mm-hmm. Great. Can affectation of the vagus nerve affect or explain post-exertional malaise, do you think?
We think so to a point, although, because the brainstem really has such a control of autonomic function and the ability that gets tied into some regulation of blood flow, that gets tied into some of the somewhat vascular associated issues with patients. So we do think there's some contribution, the vagus nerve signaling, and Mike Benalziger is a better person to talk to fully about this, to post-exertional malaise. That being said, we also think that the vasculature is likely involved. We do think that there could be issues with blood vessel and vasculature function that might further exacerbate post-exertional malaise. And there's many ways by which some of the pathogens that are can contribute or exacerbate ME/CFS cases can affect blood vessel function. Spike protein with long COVID is pretty obvious, but again, that's why we're interested in Bartonella, which infects the blood vessels often. That's why we're interested in some of these other viruses. I think Risa Pretorius' group even wrote a paper on how the herpes viruses can impact functioning of the vasculature. So I think that there could be, alongside with vagus, some other vasculature-related PEM considerations.
Great. And a question on what do you think about antibody isotypes switching from IgG1 to IgG4, enabling immune tolerance and spike persistence?
I do think that is that's possible. And, we don't even just see that research in persistence in ME/CFS and long COVID. There's actually a smart team studying pathogen persistence contributions to Alzheimer's. And they see that IgG4 class switching with cytomegalovirus when it can sometimes be found in the brain of patients with Alzheimer's disease. So there is a sort of tolerance, that's a whole other talk. But immune tolerance is definitely an important area of study in understanding why some people might not clear these pathogens and others do. And I think that understanding that the kind of immune tolerance of people's systems is an active area of research that's important.
Fantastic. We have time for a couple more. Just want to clear as many as we can. What do you think causes small fiber neuropathy in relation to autonomic dysfunction?
That's a good question. I hope that we have some more clear data on that soon, because we're running a study where we get, punch biopsies can be done. We get them from the thigh of patients. We have a group of patients. One arm is long COVID, one is post COVID ME/CFS, one is post-treatment Lyme disease syndrome, one is Ehlers-Danlos syndrome. We get the punch from the thigh, and then we send to a couple labs who are analyzing the samples. So that data will hopefully really answer this question more. But overall, one of the hypotheses we have is that, in both the blood of long COVID patients and patients with ME/CFS, we see a certain burden of what, gets called microclots. We know these from the Pretorius team, and we have another study at Harvard that's, created a microfluidics device to identify these microclots. And we see them, fibrin-related deposits in the blood of patients. We think that one possibility is that the peripheral nerves in the thigh are in the system are fed by the vasa nivorum. It's an important blood vessel, that delivers the blood supply to those small peripheral nerves. And if the vasa nivorum isn't, something blocks the ability of that to be perfused, then maybe the small fiber nerves don't get the blood supply they need to just be able to function. And we wonder if microclots or fibrin deposits might actually kind of clog up the vasa nivorum in a way that prevents signaling better or perfusion of the peripheral nerves. So that's one of the things we're looking for in these samples, is we're actually looking to see if we can find, fibrin deposits or other issues with the vasculature that feeds those small nerves.
Great. Two more questions rolled into one. Someone is asking, what test is recommended for possible Lyme? And I guess the sort of related question there would be, would you advise that people with a clinical diagnosis of long COVID or ME/CFS should get tested simultaneously for other potential covert pathogens before embarking on any treatment that may be otherwise inappropriate if they have a COVID infection that can be treated easily?
Yeah. It is a hard question because the diagnostics are not perfect. And so, any of the tests that I can tell you about are, imperfect, but I think sometimes better than nothing. So, and it really, with cases like this, it becomes an issue where someone might have to treat various infections or different kind of issues in a single case, right? So those are all considerations. But with Lyme right now, our clinic at Mount Sinai, we're using two labs. One is IGeneX, has an immunoblot test for Lyme that's FDA approved. It does seem to catch a decent amount of cases, and they have FISH testing for Bartonella and Babesia, where it's a direct detection test for the RNA in blood, and it seems fairly accurate. We also use Medical Diagnostics Laboratories, MDL. They have a Lyme6 ELISA, C6 ELISA test that kind of captures a specific peptide made by Borrelia. It can be more informative. So those are the two tests that we order that are okay. And I would recommend getting those tests. If you have an ME/CFS diagnosis and you're not sure what's driving your symptoms, if you can afford them, I don't think they'll hurt you. You just have that information.
Yeah. No, my feeling is we should be incorporating these tests into clinical trials as a baseline investigation before we proceed with any intervention. I think we're bang on time, actually. So thanks so much, Amy. That was fantastic. And thanks to all three speakers in this session. It's been a phenomenal session for me and hopefully for everyone else. And I'll pass back to Ed.
Thanks.
Thanks.
Thanks very much, David. Thanks to all our speakers. We're now going to take a 15-minute break. We'll be back at 10 to 4. Thank you very much.
Ed, are you saying anything at the beginning of this, or are we going straight in?
Sorry, what's that? Sorry.
So I'm asking Ed whether Ed needs
sorry.
to send. Are you starting this session, or we going straight in with me?
Just launch straight in, Caroline, if that's okay.
Yeah, that's fine. Okay. So welcome to the second part of this afternoon's session. So we've got two speakers this afternoon. The first speaker I'd like to introduce is Manoj Sivan. So he's an academic physician, Professor Manoj Sivan, academic physician in physical and rehab medicine at the University of Leeds and Leeds Teaching Hospital. He's particularly relevant to today's talk. He's chief investigator of two NIHR projects. There's Locomotion, which focused on long COVID, which I believe Manoj is sort of coming towards the end of Locomotion, yeah. And then there's Heritage, which I think he's going to tell us a bit more about, which is about long COVID and ME/CFS. With Locomotion, he's been instrumental in developing some of the patient-reported outcome measures that are used in long COVID clinics and some of the tests. So he's able to, as he sees patients with long COVID and with ME/CFS, he's very much able to give us the clinical perspective. So thanks very much, Manoj, and whenever you're ready to share your slides.
Great. Thank you so much, Caroline, for that introduction. So can everyone see this?
Yeah, and if you go to slideshow, yeah, perfect.
Yeah, so thank you so much for the introduction, and what a fascinating set of talks we've had just before the break. So it's very kind of really inspiring to listen to all the eminent scientists, and let's hope we find that sort of biomarker, that elusive biomarker we're all looking for. I suppose with all that excitement, I'm coming at this at a sort of a clinical end of managing patients and, the kind of issues we face in clinics and what we could be doing. NIHR has been kind of generous, as you said, Caroline, with these two projects. And hopefully what we're trying to do is raise awareness and recognition for the condition, because not to forget, when we talk about ME/CFS, we're talking about a condition which was diagnosed almost a century ago. People are working hard to get a biomarker, but we still don't have a biomarker. And if you combine long COVID, ME/CFS, we're talking about easily 3%, 2%, 3% of global population. We're talking about 300, 400 million people. So until a biomarker is found, we can't put everything on hold. We need to manage these patients. We need to work towards recognizing the condition and validation. We've not even talked about stigma, and hopefully the talk later will sort of bring some of those aspects forward, because there is still that issue we face of stigma, of recognition, validation, which are the practical aspects which millions of individuals are facing in the healthcare system. So we can't put addressing all that on hold until we find a biomarker. I know that patients strongly feel about it, but we need to look at the flip side of it, that we are failing the current patients. So we need to manage these patients as best as we can in parallel to when the scientific discovery for the biomarkers is happening. So those are my affiliations. I'm not going to go through that. And I'm going to take a very short, brief clinical look at, well, what could be the similarities and distinctions. So I'm going to take sort of two stages in the presentation. I'm going to talk about the diagnostic criteria of long COVID ME/CFS and studies which have investigated the overlap. There are lots. I've just picked sort of 3 studies, which gives you a flavor of actually what's going on in the field. And I'm going to propose a kind of a summary conceptual model of the overlap between the conditions. We can take questions at that point for that bit. Or if not, we can just go towards the end and talk about Heritage study. I'm pleased to see I'm joined here in the attendees with my co-investigators. So it's glad they're all here, so they can equally take the questions, I suppose. And then we can discuss at the end any other sort of burning questions. So it's been, we've talked about the genetics of it. We've talked about the kind of the immunology, the immune signatures. We've talked about the viral persistence and sort of the other biomarkers. But the fact of the matter is there's actually currently no single unifying biomarker for the condition, and there's no objective test. People are working on it, but there's no objective test. So you can see actually the similarities between these conditions. We know every pandemic leaves a tail of people with persistent symptoms, and we haven't actually applied the same criteria to all the patients. And I think that is one of the problems, I think, where we could have done better, that after any viral outbreak or after any pandemic, I think there should have been a unified set of clinical criteria which we apply, because otherwise we wouldn't have had this distinction of long COVID ME/CFS. Point taken that ME/CFS does have other triggers, non-infectious triggers, point taken, but Decode-ME does say that in 75% of cases, infection is a trigger. So the problem is in that diagnosis, because the diagnosis is not based on biomarker, it's a clinical diagnosis. And clinical diagnosis criteria, it can be interpreted in very different ways. And you'll see that in the next set of slides, how different it can be interpreted. We've got diagnostic criteria for long COVID. Those are the sort of bodies which have put forward diagnostic criteria. And ME/CFS is much more longer, obviously, and you have different criteria. So let's look at long COVID. It's very straightforward, really. I mean, you had WHO and you had the NICE guidance, and they do talk about impairment of daily function, but they don't specify a number of symptoms. So somebody could just have one symptom, which is persistent after, depending upon the guideline, 2 months or 3 months after the persistent for that amount of time. 12 up to 12 weeks after the infection, and you could get the diagnosis. Okay, so you could have fatigue, let's say, and if that is persistent and is affecting your daily life, it doesn't say to what extent it has to affect your daily life, you would get a diagnosis. So if you think about it, it's much more broader, it's much more liberal, and you don't have to have a PCR test, for example. Let's just deal with that, and that was because there was no testing in the first wave. So these are the issues we face. Okay, so it's a clinical diagnosis. We've got various tools to pick the symptoms up. There are about 200 symptoms described, and as Caroline mentioned, we have our own PROM which we developed called the C19-YRS, which is now getting modified to look at other conditions as well. So it is tricky how you interpret. So if a patient walks into a clinic and says, since the COVID infection, I've got fatigue, and if it's persistent for more than 3 months, whether they have PEM or not, whatever, you get the diagnosis of long COVID. Let's look at ME/CFS. So this has evolved. So from the left to the right, you can see how the criteria has evolved. And this in itself actually tells a story if you pay attention to kind of how the criteria has changed. If you just look at the time duration from 6 months, it's moved on to 3 months now. So it's trying to actually capture more patients. It's actually the time period has been relaxed to 3 months. If you look at functional impairment, it has to be substantial. If you look at the early criteria, Fuduka criteria, the CDC, the IOM, from 50% of serious functional impairment, it's getting a little bit more relaxed, and then it is required, but you don't actually specify it in NICE as to what amount of functional impairment there needs to be. New onset is interesting because they all talk about the new onset. So there is a trigger, there's a definite trigger to this condition. But let's look at symptoms. If you look at like Fuduka criteria, it was quite broad. You need to have those 5 categories. And in fact, the Holmes talks about 8 symptoms. And then if you talk, and then with time, if you look at the CDC was it still required a number of symptoms. If you look at the CDC criteria list, and then if you look at the IOM and NICE, they've dropped it really to 4 core symptoms of persistent fatigue, PEM, cognitive problems, and sleep disturbances. So if you had those 4 symptoms for 3 months after a new onset problem, and if there is a functional impairment, you do get a diagnosis of ME/CFS. So the idea is, I think, people have recognized that there are different types of ME/CFS, not just the severe types. We know about the bedbound ones. We want to capture even others who could be struggling, and the functional disability is quite a lot in those patients as well. Hence, the criteria has evolved in the condition, but you still need 4 symptoms, okay? Not like long COVID, where it's just 1 symptom where you can get the diagnosis. So moving on, let's look at the overlap. So there's a study early on in the pandemic. They looked at long COVID patients, and then they applied the ME/CFS criteria. I've picked 3 studies just to give you a flavor. So they used the 38 symptom questionnaire. They did the DSQ, which is a well-accepted questionnaire to pick up symptoms. And actually, it's very sensitive, DSQ-SF, to make a diagnosis as well. So they looked at CDC criteria and DSQ sensitivity. So, and it's a good measure. So you use that measure, and then you screen people with long COVID, and they found that 58%, sorry, there should have been a reference there. I don't know what's happened. So 58% of people actually met the criteria. So it's quite high. I mean, we're talking about 60%. And what is even more interesting is if you look at those 2 categories of people who met the criteria, who did not meet the criteria, they also had the symptoms, but they actually didn't meet the CCC criteria, sorry, because of the severity of symptoms. So if you look at, each symptom is scored out of 100, and you see the people who did not meet the CC criteria the symptom severity was less in these patients when compared to those who met the criteria, because you need to have that substantial impairment to meet the criteria. Now, if you had applied the NICE criteria, it would have been a different story, but that is 2022. So moving on, this is a 2023 German study looking at 106 participants, and they looked at they did a nice longitudinal follow-up study published in eClinical Medicine, and they look at, well, how many met the criteria. Now, again, they use the PEM cutoff as 14 hours. Now, that again, it's tricky how they arrived at that. I think there was a previous study which showed that once your PEM, the duration is for 14 hours, then you're definite to have this condition. So you could argue that again, is that true? I mean, is a PEM got to last 14 hours? So they're quite strict criteria. But in spite of that, 52% met the criteria. So these are good studies conducted in a very valid way, I would say, using defined criteria and PROMs. And you can see those 2 graphs. So the one in the green is the one with ME/CFS, and the one in the red is the one who did not meet the criteria. And you can see the difference in the PEM duration, PEM frequency, and PEM severity. So PEM is there, but it's less severe. So this again highlights the point which we need to be very careful about when people talk about distinctions and people say, well, PEM doesn't exist in long COVID. That is not right. PEM is fundamental for post-viral condition. It differs in its kind of severity and frequency. Now, this is about social functioning. As expected, you expect the red line to be higher and then the green to be much lower, because you picked people who are much more severely affected. This is, I think, this is our own study, actually. We did in 2024, and we looked at people 3 years down the line with long COVID, and we applied the NICE criteria. Now, this is again another interesting story, that when we applied the NICE criteria, we got about 43% who met the criteria. But if I just show you the table, there's another 13 patients actually who scored 3 items out of the NICE criteria. And the point they were trying to make, these are patients that they never understood properly the first point on the NICE criteria. So if you look at NICE criteria's first line, it says debilitating fatigue, which gets worse by activity. But again, there is another a double barrel to it, saying it's not caused by excessive exertion. And again, it introduces saying non-significant. They could have just called it worsened by activity, not relieved by rest, because this excessive point actually is actually what is referring to as PEM, which is point number 2. So the first criteria in its wording, I think, is a little bit confusing, and that also probably affected the kind of the prevalence we got of around 43%, if not probably would again be around 50%. So in summary, I think there are lots of other studies. I've just picked those 3 studies because they use different criteria. And this is what I think. It depends on which criteria you use. It depends on what sort of severity of patients you're choosing to include in each diagnosis. And as I said, for ME/CFS, that's quite relaxed at the moment with the NICE criteria, and it depends upon the duration of condition. So if, like we did, if you pick up people like more than 2 years down the line, and if you pick up people who are, and if you use the NICE criteria, you can see that the overlap is going to be quite substantial, probably more than 50%, I would think. If there are some tweaks, I think needs to be made to for the terminology which is used in NICE. How much more time have I got? Sorry, Caroline. Oh, I can't hear you. I think you're muted. It's fine. It's fine. I think I will move on. Because I'm not
no,
sure.
you're fine for time, and sorry.
Okay, so I don't know whether we want questions at this point, or shall I just finish heritage as well?
I think we should do the questions all together at the end. Yeah,
okay, right. Good. Okay, so just moving on. So what we're trying to do in heritage, it's an NIHR funded study, and as Caroline was mentioning, we did locomotion. It is health services research. I think it's very important to emphasize, as I did right at the beginning, we need health services research, we need advocacy for the condition, we need recognition, and we need health services. Long COVID services are shutting down right, left, and center. And ME/CFS prevalence is going up, but we've not seen a proportional increase in the number of services for ME/CFS. And you could talk about staffing levels, that it's not geared up to do research, it's not geared up to do all the kind of biomarker stuff we talked about. So those are the deficiencies. So we need advocacy in healthcare services, and I hope we did that well in locomotion. We estimated, for example, the health economics of the condition. We talked about integrated care. We talked about, we introduced and validated the lean test for dysautonomia, which is now routine practice. I do lean test on everyone with a long COVID or ME/CF now. So we've done some sort of work, sort of championing the services, and we want to extend that to ME/CFS. So this was, again, as Danny was saying, it was quite challenging. We wrote this on the sort of locomotion template. It got rejected. It got completely rejected, actually. It wasn't even like come back. But I was quite determined, actually, because I think there is a serious issue here. We're talking about at least 3-4 million people affected. You could argue that's almost like equivalent to prevalence of cancer, but there is no investment. So we made all the tweaks, and we went back in, and again there was an issue with the peer review. But finally, I mean, we got it. So I mean, that's good news. So this is what we're going to do in this project. It's again sort of health services research. We want to look at how the services are configured and what is the sort of economic burden, what is good practice. I think this is the key outputs from locomotion. We published in a series, if you remember, in BMJ, for each symptom, we published actually clinical guidelines as to how you should manage breathlessness, how you should manage fatigue, how you should manage PEM, dysautonomia. So we aim to do something similar here. With long COVID and ME/CFS under that whole umbrella of post-infectious conditions. And what we need, I highlighted that in the blue, is a national service framework. I think given the target population, if we include, like Amy was saying, all these other infections which cause post-infectious long-term conditions, we need a national service framework if it's affecting 5% of the population. We've got framework for every other condition which affects less than 1% of the population. So why we don't have a framework for something as big as this? So we're going to have 3,000 participants across the study. Those are the sites which have like different evolving models of long COVID and ME/CFS. Some are merging, some are not merging, some are shutting down. So that is not the main issue. I think what we're going to look at, well, how is having a specialist, a clinic, compared to not having a clinic, let's say. So we're going to pick a national cohort where they don't have access to any specialist clinics, and we look at their symptom burden, they look at the health economic burden, and we will talk to them, we'll interview them, we'll find out more about the issues they face on diagnosis, and we talked about the stigma, health inequalities. We're going to look at all that, hopefully. We've got a good PAG, and I think some of the members are here on this workshop as well. So thanks to them, and Elaros, I think Danny mentioned, and I think this is powerful because we're using Elaros platform. It's always good to kind of collaborate and look at whether we can all work together using some of these commonalities between the studies. So in work package 1, we're trying to understand that very diagnostic overlap I talked about. Patients of both long COVID and ME/CFS will be kind of recruited. They will all do a same set of prompts. Like we've got the YRS developed by us, and then there is the DSQ, there's FunCap, there's EQ5D, there's health economics. So all patients will complete everything. And then there's an opportunity for us to look at, well, is there an overlap on a diagnostic level, and is there an overlap at a symptom level? And hopefully we get more answers on what I was talking about. Work package 2 is around looking at the cost effectiveness of service models. I said there are different service models. There is no clinic versus clinic. So we're going to capture how much healthcare utilization there is, and how much care burden there is. We did similar work in locomotion, so we'll follow the same template, except that we didn't have a non-clinic model there. So this can tell us whether actually it's cost effective to have a clinic in the first place, or just manage the patient in primary care with GP, because there's an argument that that could be another model where we actually don't need these specialist clinics. The QIC is something which we did very well at locomotion. As I said, we published in BMJ and Lancet about how to manage those different symptoms. So hopefully we use that template. What happens is basically the clinics and experts from inside, outside the project, they meet on a regular basis. We pick a topic. Let's say, for example, severe patients. How are severe patients managed? So then the clinics will share their experience. We'll have experts from outside who will tell, well, actually, I manage the patient like this. We have people doing home visits and things like that. So hopefully we gather that rich data and come up with a consensus, because you don't need an RCT on it. All you need is consensus of good practice. And then that becomes kind of a quality indicator of how you manage service patients, severe patients. And similarly, what should be the staffing level? What investigations would you do? How would you manage dysautonomia? What prompts would you do? What is your staff to patient ratio? Something like that. So sort of data, metrics. So these then become the quality indicators of how a service should look like. And that is your national service framework, really. And then that will be rolled out, hopefully. DHSC has already backed us, and I think we are included in the delivery plan for ME/CFS. So hopefully this NSF, when it's published, could become a template for future. And if by that time, if we have a biomarker, then that's fantastic. I think that could also go in the National Service Framework. So in summary, I think, I'm trying to say, well, both are clinical syndromes, very vague criteria. I think it's interpreted in different way. You could make the overlap anywhere from 50% to probably 100%. And part of the problem is we don't have biomarkers. However, I very strongly feel all these conditions should be managed under one roof, because only then you can answer those relevant questions of what is the similarities and what is the differences. If you have separate camps, if you have separate clinics, you're not going to answer that question. And I think that's a message even to charities. I see so many charities just for ME/CFS, and I'm not sure why we need so many charities. I think we all need to come together to address this condition, and we need to start from both, like top down and bottom up, biomarker research, validity of the condition, recognition. How are we going to address the stigma? How do you answer all these questions when they say these are psychosomatic conditions, and actually it's waste looking for a biomarker? So collectively, I think we can do it better. We need services which become research centers, not just clinically managed patients. And hopefully we can come up with NSF, just not for these conditions, but all the post-infectious conditions. Thank you to the Locomotion team and all my partners. And you can drop me an email whenever you wish. Thank you.
Thanks very much, Manoj. That was great. That was really informative. We've got some questions in the chat, so I'm just going to kind of go through them. So there's a question about aspirations for Heritage, I guess. Do you think Heritage might end up recommending that long COVID and ME move away from from the rehab clinics, because most of them are focused on rehab at the moment, and come under the umbrella of another medical specialty, so maybe neurology, immunology, or rheumatology?
Yeah, sure.
So
I think Yeah, I mean, I think if there is appetite for that, that is fine. I think if there's a specialty which wants to take ownership, I think that is great. Whether we need to move away from rehab, I think I will strongly decline that, because if you think about
they don't say move away. I think
because if you think about any condition, let's say you've had a stroke, or if you have a brain injury, rehab starts right from day one when you're in ICU. So if you don't have rehab, you'll end up with a completely disabled population, irrespective of what medical condition they have, whether they have cardiac MI, or you just can't, treat the stroke and leave the patient. Who's going to rehab the patient? So I think I've heard this a lot in patient forums. I think we need to show more respect towards professionals who are managing these patients. And all we are trying to do is we are trying to even put off the people who are currently looking after patients. I think that is totally not good. I think we need to really address this and hit it on its head, that we don't have this discussion again.
Actually, the second part of this question just makes the observation that actually some of the and I don't think they're being negative about this, the innovation in long COVID treatment has come within rehab. And I think they make the point that if there was a medical speciality that adopted it, that might be, as you say, the opportunity to do more research on the more medical side to complement the innovation that has been taking place in the rehab.
Yeah, true. I mean, my argument is, well, rehab medicine is a medical specialty. Rehab medics go through medical training. So if people have these misconceptions that rehab medics are physiotherapists, I think that again is a misconception, because they go through a very rigorous medical training. Well, they could do research. That's not a problem.
So then somebody else says, following on from what you were saying about doing the lean test, do all the clinics do the lean test? And if and therefore can long COVID and ME services prescribe drugs if POTS is diagnosed?
Absolutely. I think we've done quite a lot of work on this. And so people have to look at our publications. So lean test is as good as a TIL test. And I think clinics need to have the expertise to prescribe medications based on the findings of the lean test. And I think I completely back the argument that we need medics in these clinics to manage dysautonomia and to prescribe medications. And it actually saves quite a lot. You're up against cardiologists who've got long list for TIL tests for various other conditions. And you can't do, if you're talking about 3 million population, you can't be TIL testing 3 million. There's studies showing TIL test and lean tests are equivalent. So it's a simple test, can be done in clinic, gives you all the relevant information you need to treat the patient pharmacologically and even non-pharmacologically.
Then someone's picked up on the fact that you seem to be using different PROMs for long COVID versus ME/CFS. So you're using the YRS and the DSQ for long COVID, but the YRS and FunCap for ME. So they just queried, was that the case?
Oh, yeah. So I think
why is that?
Yeah. So I think I've just listed it that way. So these are the PROMs which have been validated in these conditions. But essentially, every patient in Heritage will be doing all the PROMs. And I think this is a good opportunity also to understand how each of these patients sort of score on each of these PROMs, and also to cross-validate PROMs in other conditions.
And somebody asked, could you clarify what Eleros is? Just tell us who Eleros is.
So Eleros is a digital company, and they have a platform of PROMs, basically. And it can either work on a standalone way that patient enters data or their symptoms into the app, and it gives you a score for how severe your symptom is or how severe your functional disability is, and you can track it. You can use for your own monitoring, and you can. So that's a standalone facility, and then it's like a paired facility with clinics where the clinician can actually communicate to the patient, because it's very a dynamic way of patient completing the PROMs, and then the clinician in the clinic able to see that and then to kind of provide advice and interact So, and it cuts down on all the paper and all the admin. So it's a digital PROM. It can be used it's used in clinical services, and it's used in research. And as you can see, it's used in heritage, and it's used in Danny's study. So that's what the platform is.
Okay, and I think just one last question, and then we'll move on to the next presentation. So, this question about severe ME CFS, so the 25% of people who have severe ME, how are you looking to administer your tests and PROMs with that group? Are they included in
Yeah, they are
project?
definitely included, and I think we've made a number of provisions that the PROM can be completed by patients' relatives, or even the clinician can complete it in conversation with the patient. They can take adequate breaks. They don't have to do all at one go, because the whole battery of PROMs is quite extensive. They don't have to do it at one go. They can take, I think we said 2 days to complete PROMs. Yeah. So yeah, I mean, we've got a very highly intelligent PAG, and they've closely looked at all this, and they have suggested measures we need to be mindful of when dealing with these patients.
Fantastic. Thanks so much, Manoj. That was really fantastic presentation. So next we have Nisreen Alwan. So Nisreen is head of the Centre of Population Health Studies, and she's Professor of Public Health at the University of Southampton. She does research on how to prevent disease and optimize well-being of young families, looking to better long-term health, and particularly a focus on health inequalities. So she has a reputation across the public health response to the pandemic. I'm sure a lot of the people who are listening today or watching today already are familiar with her work, particularly focusing on the recognition and quantification of morbidity from COVID-19, and particularly focusing on the call to count long COVID. So she's particularly carrying on with her work looking at inequalities in prevention, recognition and care. So thanks very much, Nisreen, and if you're okay to share your slides.
Thank you so much. Caroline, can you see my slides in full screen?
Yes, we can, yes.
Excellent. Okay, thank you so much for that introduction, Caroline. And thank you to Manoj, because I don't think I would have had any. It's a perfect introduction or a talk preceding my talk. And in fact, I've got a one slide comment, but anyway, he explained it better, so I can skip through it and save a bit of time. But I wanted to just talk about the case definition aspect in shaping research and shaping how health inequalities are narrowed or widened for ME/CFS and also for long COVID. So so I wanted to start by really trying. This is the last talk of the day. I don't know how many people are still there already, still listening, but I thought this is something to kind of might interest you. We a few years ago, we did a systematic review of the prevalence of long COVID, and there have been a few systematic reviews around the prevalence and many studies. It was lots of work, and I believe a couple of my co-authors, thank you to them, they're probably in the audience, I don't know if they're still there, but basically it was a lot of studies. We include 130 papers, and the prevalence, as you would expect, was very, very kind of wide. The range was very wide, the overall prevalence, but a very striking findings that we found is actually a lot, it kind of partly dependent on how you assessed long COVID. Now it's not probably a surprise to people, but really when you look at it this way, so studies using healthcare records diagnostic coding had a pooled prevalence estimate of 13.6%. Those that were using self-reporting of long COVID had a pooled prevalence estimate of about 44%, massive difference. But actually, the studies that did some testing, like, they did a scan or respiratory function or some sort of test that looked at pathology, had an even higher, a pooled estimate of about 52%. So really, that was the method of assessment of what long COVID was and how it was sort of assessed. Was directly linked to kind of the prevalence estimate. So it's really important to understand how we assess these conditions. So why is the case definition important? So obviously the case definition is important because it identifies somebody with the condition, and that determines the receipt of healthcare support, treatment, rehabilitation, if you're eligible for adjustments or benefits or occupational recompensation, all really depends on your case whether you meet or not meet the criteria for case definition. And obviously, also, it determines who gets included in and who gets excluded from criteria to join research studies. And from my perspective in public health, it's really important because it's about the quantification of population disease burden and how we can plan services and how we can plan preventive services as well. So I'm really interested in it, and I was interested in it right from the start. This was the first BMJ webinar on long COVID in early September 2020, and they asked me the first question. I was like, "Oh, how do you define it?" And I said, "Well, it's just not getting better. Basically, it's not recovering for weeks or months after you start the symptoms, whether you were tested or not, because many of us were not tested, as you know." And so that was a simple sort of definition to start with, and I continued to be interested. And then I sort of went on and was thinking about, well, let's kind of define it by recovery. So if you can kind of define recovery in terms of how long you've been well after COVID, and what sort of symptoms, you can kind of, define it by what it's not, basically. That was, a simplistic approach, and we know now it's very difficult, and as Manoj, kind of touched on in terms of kind of what defines long COVID and how people can get included in, be recognized. So there have been many guidelines around sort of many kind of definitions, WHO definitions, sort of, as you can see, there are some CDC definitions, others, and they kind of use slightly different criteria. This is the one that Manoj shared, and so I won't talk through it in terms of the kind of the diagnostic, the detailed criteria for ME/CFS diagnosis. And as Manoj pointed out, really it varies in terms of the duration, in terms of what symptoms are included, in terms of the whether it requires a certain functional disability or not. So all of these things, you can see who can be defined as having a condition obviously will differ by using which criteria. So I thought it'll be, take you on a little bit of a journey just to consider and reflect. My talk is really mainly about kind of reflecting around this, using some examples from some of the research we did. So what's in a case definition? So the case definition, I think the first thing that what's the purpose of the definition? I had it at the end, but then I moved it to the first criteria because I do think that's probably the most important. What are we using the case definition for? So to my mind, case definition could be, serve one of three main purposes. One is that you are trying to define who gets included in research and who doesn't, and that obviously defines who kind of, how it shapes your findings, but it depends on the cohort of people who are included in your research, whether you're a very specific cohort of people, whether it's a broad, a group of people based on your criteria. Then obviously there's the other, the purpose of the count long COVID bit, or, what's the prevalence of ME? That's a big question, what is, the population health burden of, these conditions. So this is a bit about public health systems or surveillance. And then we've got the very important function of clinical case definition, where you need to have to get the diagnosis, because that's your gateway to care and treatment and rehabilitation. And I put health inequalities there because that's really important, because that kind of links in terms of kind of the population health, who gets, more burden of the condition, or more burden of not getting their sort of health condition addressed and treated and cared for. And just thought I'll just kind of very quickly say, well, what we mean by health inequalities or health inequities is systematic differences in health status in a population, mainly arising from the social conditions in which people are born, grow, live, work, and age. So that includes things, demographic characteristics as well, age, gender, ethnicity. And there is a value judgment in that they are unfair and they could be reduced by the right mix of government policies. So just going back to those criteria, when I said what's in a case definition, the next bit is what is it? And basically what is it is, again, Manoj touched on this, is the symptoms, ME/CFS. Some definitions have PEM, some don't. Some have specific symptoms, some don't. Long COVID, some definitions have number of symptoms, 1, 2, or 3 symptoms, etc. So that's really kind of it's variable. I'm putting this slide because that was one of the first surveys we did of long COVID. These are people who are self-identifying as having long COVID, and there was a variety, obviously, of symptoms. And at the time, we were really interested to look at the main differences in the symptoms between people who had a lab confirmation of COVID and who hadn't. But actually, if you include all of these symptoms, and there have been more, loads of symptoms for long COVID, sort of recognize, this is a kind of a broad definition. So how do you narrow it down if you want to look at sort of mechanisms, etc., because people with different sets of symptoms might have different mechanisms. The other component is for how long have you had it, how long after the cause, if it was an infection, whether it comes and goes, is it relapsing, is it fluctuating, and whether there is a recovery or remission phase. And I have to say, most of the examples I use are mainly from long COVID. I do think these are the similarities, but I use them because obviously that's been where my research has been on. So this is the how long bit. It's complicated. I mean, for long COVID, there are similarities to ME/CFS as well, is that, the pattern is different. So this is the survey at the start where we had people describing their pattern as relapsing, comes and goes, or fluctuating, sort of up and down, never goes away, or it's a constant. And then we followed up those people about a year after to ask them about the symptoms. And you can see here that, some of these patterns change, so the fluctuation could become relapsing or could become constant, etc. And that complicates the picture because it depends on when you're asking them, if you're asking them for research, where they are in terms of their symptoms. Are they in remission? Are they in sort of having different symptoms at that time? So if we really keep, so this is the thought, so if we really keep the definition broad to accommodate the different patterns and phenotypes, could we be diluting the research findings and pushing them towards a null? Because we want to be a broad church. We want to kind of include people, definitely in terms of accessing clinical care. But in terms of research, are we risking diluting the finding because we are including different subtypes of phenotypes who may have different mechanisms and may respond to different types of treatment? And if we don't do that, what do we just call them subtypes? Do we kind of start separating these conditions a bit more within ME/CFS, within long COVID, etc.? Now, obviously, we're trying to put things under the banner of post-infectious diseases, everything. So how do we kind of look at, such classification? And when long COVID and ME, if they are in effect an exclusion diagnosis, the variety of symptoms we have means that we may clinically, the doctors and are trying to exclude other things, but such exclusion processes are not standardized. Some of them are much more kind of detailed and thorough than others, and that will also, kind of result in disparities in terms of who gets the diagnosis. And how do we deal with overlapping conditions that present similarly? So, if we only, because when we're trying to, one of the things about kind of long COVID and ME, you're trying to exclude other conditions, but obviously we have a population that's growing in multiple long-term conditions. The likelihood is that people would have the other conditions. They could also have long COVID or ME/CFS. And, the key thing is not to kind of try and separate the two, is actually how can you define these conditions in the presence of other conditions? So just going back on the impact bit, also as Manoj said, depends on, if that's in the definition or not. The WHO definition of long COVID has that, and that it impacts everyday activity. We know that a lot of people are impacted. This is from the latest, ONS prevalence data on long COVID, and we see that about 1.5 million people had limited day-to-day activity, and about 380,000 had, in the UK were limited a lot. So that's how they identified, their impact. And also there's that's related to kind of the notion of severity in the case definition, how much, in that survey, that in the follow-up survey that I mentioned, where we followed up people, we looked at indicators of severity, impact on jobs. So we looked at job loss. Sorry, this is a bit small, but this is saying job loss due to to having long COVID, and whether that's changed over time, and there's not much change in that. And the other bits is loss of income. Again, a proportion of people said yes, agreeing here. And that seemed to more or less stay stable. So do we include those dimensions in the case definition? So yes, so this is a question. Do we include an element of impact on daily life? Do we include measures of disability, well-being, and work? And then what causes it? No, obviously for long COVID, there's more about waste. We know that COVID causes it. And for ME/CFS, obviously there are many. We heard earlier about kind of the different potential causes and mechanisms. I won't dwell too much on that, but that could be part of the definition. And then again, in the early days of the pandemic, I thought a lot about that, and we tried to establish some criteria, not just to define long COVID, but to define the infection in the absence of definitive testing. So that you're trying to establish the cause. So going back, why did I go back? I went back to this to remind you of those differences in that we found in the review. And so we found that those who are coded in the clinical records had kind of much lower prevalence. So if we use clinical diagnosis as a case definition for research, what role do things like postcode lottery in terms of who gets a diagnosis, what's the knowledge of the kind of, healthcare professionals diagnosing and giving that label, and also what role does stigma play here? So stigma is really an important aspect in getting the label, if you like, in getting the diagnosis. So we asked people as part of the follow-up survey about stigma, and we looked at 3 dimensions. One was looking at people who have enacted stigma, so they've experienced discrimination because they've related to having long COVID or declaring that they have long COVID or symptoms of long COVID. And then there's internalized stigma. People feel embarrassed, feel tainted, feel different from other people. And then the third dimension is anticipated stigma. So people then hesitate to talk about their long COVID or seek services because they think long COVID, people think they fear that stigma of people thinking it's not real, it's weakness, etc. And we found very high levels of stigma. So we found that, you can see, those who said sometimes or more often in terms of experiencing stigma, all of them are very high, both enacted, internalized, anticipated. And we found that those who had a clinical diagnosis, met the case definition, whatever it is, had slightly higher level of, it felt slightly higher level of stigma. And perhaps that is because they maybe talked more about long COVID in order to get that label. About third people regretted having told others about long COVID. About 2/3 said they were very careful who they tell about that they have long COVID. So really high levels of stigma. That determines who actually gets kind of recognized. This is unpublished data from the English GP patient survey data, which is data from a national survey, and you can see that people that the stigma as well as kind of how people kind of interact with having the condition varies by their characteristics. So an example is like we found that males were more likely to be confident to manage their long COVID, but they're less likely to have a conversation with their healthcare professionals. Also, people who were, age, and there's a variation by age, there's variation by ethnicity in terms of how people kind of talk and have these conversations about their COVID, which will obviously lead to them having the diagnosis. So it's really a complex picture. It's not just applying criteria for case definition. There are layers of what we call epistemic injustice here. So there are layers of testimonial injustice, which is about who you are as a person with your characteristic, determines whether your story is believed or not about yourself. This is your story about yourself. This is what we call testimonial injustice. So this is about who you are. And then the hermeneutical injustice is about really, the condition. How is there a framework to talk about your condition, which is ME/CFS or long COVID? Is there an established framework, like, for example, there is for things like diabetes? And all of these things shape how the case definition, gets applied to you as a patient. So it's not uniformly applied based on the criteria. very last thing, 2 minutes, I want to touch on self-reporting, because self-reporting, as we saw, had a higher prevalence, but actually, but we find that those, you know, in the review, those ascertaining pathology had even a higher prevalence. So self-report data get routinely labeled as subjective. They get routinely considered more biased than objective data. And the assumption is that people misremember or exaggerate or misinterpret their symptoms. This is part of the epistemic injustice as well, that you don't even know the truth about your own story. So blind following of this sort of traditional framework can lead to stigma and injustice and inequalities, particularly when we have conditions. There's no diagnostic test, so the clinical diagnosis is variable, it's non-standardized. This framing is not justified. We don't have a magic test. We've heard all about this. We don't have that magic biomarker yet. So we can develop criteria to help standardize or triangulate self-reporting for inclusion and research. And this is something that Decode ME, for example, did, where actually there were some comprehensive criteria for joining the study. You know, but that joining was kind of recruitment was from the community, so it was reported by the participants. And I've wrote a little piece about this recently, and I thought people, if we can just stop a minute before we pass this judgment and ask ourselves whether, before judging self-reported data of inferior quality, can it be measured using an alternative way? You know, is that alternative way a better way? Is there a magic biomarker that we can use? What exactly makes that better? You know, if we say, we want clinical coding. Well, why do you think clinical coding of such variable quality, affected by all these, you know, kind of factors around, you know, the stigma and the inequalities and the vague and broad definitions? Why do you think it's better than a kind of a thoughtful way of self-reporting through questionnaires, for example? And can certain groups of people be disadvantaged by using that other alternative way, which is, for example, you know, coding, clinical coding, and to use for research inclusion? And can the alternative way be combined with self-reporting to strengthen the data? So what are my conclusions? I guess my main thing was just to get you to reflect a bit that this is not a straightforward thing. So definitions can vary by purpose. You can have it from the research, for clinical care, for surveillance, and a one-size-fits-all approach is too simplistic, really, for these conditions, long COVID and ME/CFS. Case definitions can be applied, so the same case definitions can be applied unequally, and that's influenced by the patient's characteristics. So we need to research on how to construct fit for purpose case definitions for long COVID and ME. So we need the case definitions for research inclusion, but we actually need research to be better at doing case definitions. And we need to raise awareness between, you know, healthcare professionals, researchers, and research funders, because I just always find this very simplistic, uncritical approach to case definitions I come across, you know, whether it's like research peer review or sort of, buddies or health professionals. I think we need to raise awareness about this, really, and get people to reflect, because we need those valid and reliable case definitions for meaningful and generalizable research and to reduce health inequalities. Thank you.
Thank you so much, Nisreen. That's fantastic. We've got some questions in the chat, so I will go through some of them. So did your research show, or did you find in your research that people with long COVID could get progressively worse, not just better?
Yeah, I mean, it really depends. We ask questions about the pattern and about the indicators like job loss and loss of income. So if you use some of these indicators, yes, some people can, you know, so some of them went from sort of fluctuating to constant. Yes, some people get worse, some people get better. What we did find is that recovery, people who said they completely recovered with a very small minority, maybe if I recall correctly, it's about 5%. And I think that's something I'm really interested in now, is how, going but full circle to the start, how do we define recovery from long COVID? Because most people, and I think that is another similarity with ME/CFS, most people may describe themselves as in remission, maybe, or particularly if you have it for a long time, you're very hesitant to kind of say, no, that's it, I've recovered. You know that it might come back or in a different way.
Then there's a question which I think is very relevant to your discussion about stigma. So this is aimed at the panel really, but I'm going to ask you, Nasreen, what you think. Would you like to comment about, I think is it Suzanne O'Sullivan, O'Sullivan, who has been talking on the BBC about long COVID and saying long COVID is primarily a social media meme. So the question is, do you agree this is deeply unhelpful? I think you're going to say yes. But how can we counteract this basically completely ignorant somebody who's going around saying things that are just so ignorant?
Yes, well, thank you for that question. It's interesting because I got told that I am mentioned more than once in that chapter. So I need to look into it a bit further. But obviously, just generally, so I haven't actually read the book or the chapter yet. So, no. But it's interesting you asked me this question. I was just been thinking about it yesterday. We had a conversation about it. So, yeah, I mean. I think a critical approach. I mean, I think a different or critical approach to thinking what I was saying now. Well, how do we define a condition?
Yeah, sure.
Valid, but actually, over the years there have been these approaches where you're not really sensitive or aware of the context of the massive dismissal, and I mean the levels of stigma is so high, people don't want to talk about it. And I think it's not fair not to be sensitive to this, even if you have a different perspective, and to bring it to kind of discussion.
Yeah,
I think more to say about this, but I just need to look into it a bit more.
I don't think that saying it's primarily a social media meme is a particularly academic approach to that kind of incisive discussion. When do you think the NICE guidelines on long COVID will finally be completed and published? Do you have any information on that?
I don't know. I don't, I'm afraid. So I don't know. And I do think that, I don't know. Hopefully, maybe other people are. I don't know. Manager's still here and he knows, or anybody else. Yeah.
I think there were some discussions, Nisreen. Not sure about the timelines, but there is some discussions and people are being contacted. So I don't know the timelines, Caroline.
Okay, people saying thank you for all the talks today. There are many congratulations to the organisers. Nisreen, I just wanted to kind of tie in a bit with what Manoj said and what you've said about obviously not having any biomarkers, even for long COVID or for ME/CFS, and then not even definitely not having biomarkers for the subgroups that you discussed. And I wondered, how confident do you think we are therefore in defining those subgroups within, by questionnaires or by just clinical expertise in the clinics? And because obviously the different how you treat somebody, which subgroup they're in, and it really affects how your treatment strategy.
Yeah, I do think that the first, I think there's again one of the very different, sensitive balances is about who to include in different research studies, and I think sometimes that's what I'm arguing now is that we can have a more inclusive clinical case definitions. We're not sort of discriminating in terms of who gets care and support. We need to be sensitive to that. But I think for research study, we may need more specific inclusion criteria case definitions, because it looks like a lot of these some of the research, as I said earlier, you don't pick up the findings. There's a lot of noise in terms of kind of the different groups. And I think we can do that. I think it is about being explicit about it, perhaps, for research studies, and research studies saying we're only going to recruit this group. That doesn't mean this is all of long COVID or ME/CFS, but we're interested in this group, and then we kind of work a bit more on refining those criteria.
Yeah. Okay. Somebody's asked about the top treatments that help people with ME to go into remission, but I'm not sure we covered that.
I'm not really qualified to answer that.
No. If anyone has the answer to that, please let us know. That would be really, that's the golden question, isn't it, that we want the answer to. And then somebody's mentioned as well the upcoming, again on stigma, the upcoming Royal Society of Psychiatry conference, where there's some people speaking on long COVID that perhaps have different views, that have views about it being psychosomatic.
Yeah, and I think again, I'm not going to be tiring of raising that this needs to be really. I don't think we need to shut down different opinions. I think these opinions need to be sensitive to the reality. I think sometimes they're detached from the reality of what people are struggling with in terms of just accessing recognition and kind of basic care for their conditions, because it is very hard to talk about long COVID, and it's hard, and ME/CFS. But I mean, I've got the lived experience for long COVID. I am, as somebody who studied it, I'm a professor at a university. You think I'm in a privileged position, but it's hard. I mean, I'm also a woman from ethnic minority. All of these things interact. It's hard to actually kind of still to talk about it. The dismissal is I mean, there's less dismissal because I think less of like, oh, it's not real. But I think there is wide dismissal of like, there's nothing to be done about it. The research we did, actually, the bit where I showed the epistemic justice, was actually recruiting people who've not got a clinical diagnosis. We recruited from the community and we established some criteria. And the amount of people have a lot of self-doubt. They feel like long COVID is not even deserving of you going and speaking about it with your doctor. There are more important things. I mean, this is what people are thinking. And this is not helpful. We're really kind of widening that. So you need very high health literacy, very high confidence, really. Or you need to really have struggled a lot in the system in order to get your basic level of kind of support and care in general. So, yeah, I think we need to tell those who are not sort of aware or sensitive to this, we need to keep repeating that. Manoj wants to say something.
Yeah. No. On that note, I think I would like to add that I think the way we're going to address is absolutely by doing basic science research, which is great, but we are not at that point where we've made that breakthrough of finding the biomarker. So I think what we could do clinically, though, is with all the patients, number of them are here, and the limited amount of clinicians we have, is to join forces, really. And I think I've told enough about this in my presentation. I think partly the part of the problem is we have this different terminology. We have long COVID, we have ME/CFS, we had post-SARS, we had post-MERS. So what's happening after each major outbreak is these conditions are getting isolated. And then, well, we know in about 10 years, there will be another pandemic, and that will be called, I don't know, post-Z syndrome. So with these kind of pockets of patients getting isolated, we can't move the field forward. We will move the field forward by bringing everyone under one umbrella, having a common terminology, so that in every pandemic or every viral outbreak, we use the same terminology, whether we want to call it post-infectious conditions, whether we want to call it, like the society, the clinical society, we call it IPIC. So we need terminology which is common, and so that then with subsequent outbreaks, there will be more recognition. And then your biomarker research in terms of the subtypes, it can pick up those subtypes. Actually, even more better if all these conditions are under one umbrella. So I think there's a work to be done on the clinical. Forget about the biomarker research, even on the clinical side, in terms of case definition, what Nesreen referring to, there's some work to be done. How would we come up with a uniform name which could be applied for these conditions?
Actually, I'm going to reframe that question about going into remission, because I think what the question's getting at is not, I didn't mean to be flippant about it. I think what the question's getting at is, did you see any characteristics or treatments that predicted remission at all in your data, either of you? So the question is, what are the top treatments that have helped people into remission? But I think that
if it is very specific to ME/CFS, I mean, I've seen a number of people in remission, and there are a number of treatments which worked. Some of non-pharmacological, some are pharmacological. But it's very difficult to actually, point to one particular treatment which worked in everyone. So few people talk about naltrexone working, whereas at the same time, it does nothing in someone else. People talk about, vagal nerve stimulation. It works, some it doesn't. But we've certainly seen patients go into remission and, perform very well. But there's not one treatment I could pick up to say it works for everyone.
Great, thank you. Okay, I think we've run out of time, and so I think I'm going to hand over to Ed to just wind things up.
Thank you very much, Caroline, and thank you to all our speakers today. It's been a fascinating and insightful afternoon. It really has. Just to say that let's not stop the discussions here. Prime is all about making connections and continuing those discussions. So if you do have unanswered questions or would like to make connections, then please do send an email to the Prime team. So that's prime.partners@ed.ac.uk. I'll put that into the chat once again. And also the fact that this has been recorded. So the recording will be out in the next couple of weeks, and that'll be promoted through the Action for ME website. Our next webinar is going to be on the theme of post-exertional malaise, or PEM. So that's going to be on Thursday, the 30th of July. And again, that will be promoted. The program will be up on the Action for ME website and also LinkedIn as soon as we can. And the other date for awareness is our first Prime Symposium, which is going to be in Edinburgh on the 28th and 29th of September. That is going to be a hybrid event, so there'll be an opportunity to join online as well. And again, registration details and the program will be put up in the next few weeks via the Action for Me website as well. So that's just about it. I just want to thank our speakers again, thank our chairs, but most importantly, thank you all for attending. It's been a fantastic turnout, and please do come along to our next webinar in July. Thank you very much indeed.